within the so this is a nice cartoon showing how or where all those look at. So if you look at the biofilm structure, if these are the cells, and you can clearly see they are secreting all these different types of molecules. So if you look at the clue, and this is the exopolymeric substance, right? And then also polysaccharides, protein, eDNA, lipids, and a lot of small molecules. So it's just a cartoon showing all of those components, part of the biofilm structure, they are localized within the biofilm. And so this is also another picture. Again, you will see this. These are the cells, and these are the molecules inside. Okay, and I think by staining for individual molecules like a carbohydrate or polysaccharide, or you are able to differentiate the different molecules within the biofilm matrix. Okay, and essentially this is what staining methods they use to localize these different molecules within the biofilm matrix. Okay, so just to show you that if you consider these polysaccharides, and these tables are long tables, when you get this, you go over the slide, the material, to find out what are the specific molecules or type of polysaccharides made by individual microorganisms. Okay, and again, for me, I don't want to go over the slide in detail to tell you what are those molecules. So, for example, if you look at Bacillus subtilis, it produces this exopolysaccharide, okay, and the location is extracellular. Of course, they are making this, right, and it's excreted outside, so we call it extracellular, and the function of this molecule is adhesion, scaffolding, and stability. Of course, it helps the bacteria to attach to the surface, adhesion, right, and then scaffolding. The scaffolding is the scaffold, the structure of the biofilm, and then also it contributes to the stability of the biofilm. Okay, this is an example for this organism. So same way, if you look at the Staphylococcus aureus, Streptococcus mutans, Pseudomonas, Vibrio, or Candida, you can see all those different types of sugars or the carbohydrate are made or produced by this organism. Strep mutans called glucans or fructans is pretty sticky in our mouth. Strep mutans cause dental caries, dental infection, and you can clearly see this type of carbohydrate is made. Staphylococcus aureus also makes varieties of molecule like polysaccharide intercellular adhesion called PIA. This is a long carbohydrate, and it's extracellular. Again, the function is very similar: adhesion, scaffolding, and then attachment, and helps protect against the antibiotics. So all of the specific function the carbohydrate play for biofilm produced by individual organism. Everybody clear? I'm just using some example. And if you are taking individual pathogens and these molecules, and essentially we do studying in the laboratory, you can genetically knock the gene, right? And I think we're missing time to time. Oh, battery. Okay. Turn it off. Okay. Can you hear me now? I think it's much better. Yeah. There are more seats in the front. Come on over. Okay, you are all settled now? Can I start? Okay. You missed what I said in the beginning. That's okay. You can catch up from them. All right. So we are from Max. Okay, the polysaccharide molecules produced by individual microorganisms and how they contribute to biofilm formation, right? And so, as I said before, or indicating again, is that if you want to study the role of specific molecules contribution to the biofilm formation, genetically you can modify it, right? You can knock out that gene or delete that gene and see if this gene or the gene products contribute to the biofilm formation. And so the studies in the laboratory, this is how we do it. So PSL, L or alginate. So you can knock out these genes in this organism, for example in Pseudomonas, then look at this organism's ability to biofilm, okay, or also ability to produce carbohydrate, the polysaccharide. And you can analyze it by analytical tool, using analytical tool, and also functionally you can also do the assay in the laboratory setup, see if they're forming biofilm. Sometimes they may require multiple factors, but if you delete one factor, then the type of biofilm would be much weaker than the wild type, which has all the genes. You will follow me? This is how we generally studied when you were doing research. So same way Vibrio also use certain carbohydrates, also use these molecules for biofilm formation. And in terms of function, if you read this, again I am not going to read all of them, is that they are very general function. They help the bacteria or the microbes to attach, which is the adhesion function, right? They also provide the architecture of the biofilm. We talked about yesterday, different layers, right? That architecture is very important. And also the ability to stability of the biofilm, okay? And then we will study more is that lot of these carbohydrates help absorb antibiotics or antimicrobials or sanitizers used to inactivate microorganisms. In general, if you apply some chemicals or antibiotics, so this carbohydrate goes, right? If they absorb these antimicrobial molecules, they are not going to reach the bacteria. Do you see this? And this is how the cells are protected inside the biofilm. Even though those cells are sensitive to antimicrobials and so that's one of the issues. This is the same problem. Okay. Oh, they cannot hear me. Oh, okay. 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 So this is all those different functions attributed to these polysaccharides. Not only these antimicrobials, also it can absorb all those other materials we talked about. Okay, and then there could be also food for the microbes because this is carbohydrate, this is sugar, right, and rich in calorie or rich in energy, and so the microorganisms can receive a lot of these molecules or materials from these carbohydrates. Okay, so this is one main molecule. Then talk about the proteins. We talked about the composition of biofilm matrix. Carbohydrate, those who came late, carbohydrate, protein, right, eDNA, extracellular DNA, then lipid, and then there are a lot of molecules, right, different types of molecules secreted by the microbes. So they are the matrix component. We call it matrixome, right, large number of molecules, part of the biofilm structure. So we just finished talked about carbohydrate. Now just look at proteins. Okay, so the proteins, for example, Bacillus subtilis again, and it produces some type of proteins, and these proteins play important role. These are extracellular. Again, if you look at their function, scaffolding, cell to cell binding. It's the cell to cell interaction can be mediated by proteins. Okay, these proteins are secreted by bacteria or microbes. They also, some bacteria produce flagella. We talked about flagella yesterday, right? Flagella is very important molecule or structure, help the microbes move or swim, right? So depending on which environment they are, they could use the flagellum. They could use the flagellum to arrive on the site, as well as they help them to attach to the surface. Okay, and then what happens with the flagellum? I think I'll show you some additional pictures. So the bacteria, once they start forming biofilm, they don't synthesize flagella anymore. They turn off that gene because there is no need for flagella when they are inside the biofilm. Everybody got it? So they need the flagella to get there. Once you arrive there, then I no longer need my flagella. See this? So they can turn off. They will use that energy, they can direct that resource. Instead of synthesizing flagella, they will produce a polysaccharide or protein or other molecules. You will see that? And this is how the transition happens within these cells. When they are becoming a sessile cell, these are the genetic difference happens. But when they need to be planktonic, right, they need to have their flagella synthesized again. So they can turn on that gene so that they can synthesize flagella. Everybody clear? All this transition going to happen within the biofilm or biofilm producing cells. Okay, so there are a large number of proteins are produced by different microbes that help them to achieve some of these goals. Staphylococcus aureus, we know that this is one of the notorious biofilm forming bacterium, right? And we know a lot about it, and still there are a lot of room to learn, like fibronectin binding protein, for example, FNBP. Fibronectin binding protein, many pathogens actually make that protein, but this is Staph aureus, it makes one of this protein, it binds to the fibronectin in cells. So in our cells, we have fibronectin molecule, okay? And then Staph aureus makes fibronectin binding protein, which interacts with the fibronectin in the cell. And once they colonize, they can now continue to form the biofilm. Everybody clear on that? And so, because they do a lot of skin infection or skin related infections, and this is going to be very important factor for them. Then you will see also staphylococcal protein A, SPA, Staph aureus surface protein G, also biofilm associated protein called BAP, BAP. BAP is one of the proteins made by many pathogens, not only Staph aureus, you will see also other pathogens, they also produce BAP. Okay, and so that become an important biofilm structure or biofilm protein. And then also phenol soluble modulins. So these are number of proteins, and if you look at their function or location and then the function, they are very similar. Okay, and generally there will be minor difference between proteins or among proteins from different organisms, but overall you can see the adhesion, you can see this common, right? Cell to cell binding common, adhesion common, and scaffolding, stability, a lot of these functions are common. And then some proteins also called immune evasion. They know how to evade immune system, otherwise immune system can come and destroy the organisms. And so they could have also pro-inflammatory response. We'll talk some of those things later on. And so these type of functions are essential for this organism from the protein standpoint to form biofilm, okay? And eventually that biofilm, depending on what they are forming, that can be part of the disease process. Is it clear? Okay. Same way, if you look at the strep mutans also produces large number of proteins. Okay. We're going to have next lecture on the streptococcus. Hopefully there will be some proteins she will discuss about how they contribute to the formation of biofilm. Okay. So this way you can find out this large number of proteins produced by different microorganisms. They are used for not only attaching the epithelial surface or other surfaces, biotic or abiotic, but also they are contributing to the pathogenesis of those organisms. So that's why these proteins are extremely important. Okay. All right. So you can see the Pseudomonas, Vibrio cholera, Candida. Again, you can go over all of these individual proteins. You see this one, Vibrio cholera also produce BAP protein, biofilm associated protein similar to Staph aureus. So many organisms produces different type of biofilm forming proteins. This one is called mannose sensitive hemagglutinin. Flagellum, we mentioned about the flagella is one of the structure. So on, and then you can see the functions are very similar. Okay. So after this, now we're going to talk a little bit about the other molecules, right? The nucleic acid. And if you look at this, this is wide distribution in bacteria, archaea, and fungi. Nucleic acid means any cells has nucleic acid, right? There is no specificity. That means once the cells in the community, some of them are dying or dead due to whatever interaction they have, or sanitizers, or some cells are dying because of the starvation, right? We don't have enough nutrients, and quorum sensing, lot of factors can kill, because this is a natural process of turnover. Some cells will survive, some going to die, and those dead cells are going to release their DNA. And that's the DNA we are talking about, extracellular DNA, or called eDNA. And that could be chromosomal, plasmid, any type of nucleic acid. And if you look at that, we call it extracellular. So if you are studying biofilm, if you are an organism making biofilm, one of the properties would be they should release eDNA. Correct? This is one of the things, right? For example, carbohydrates. They will be making those specific carbohydrates. They should be making those specific proteins, right? So you can analyze them in the laboratory. So eDNA analysis would be very important, right? So you grow the cells under biofilm forming conditions, and then if they are extracellular, we are not looking for DNA which is inside, right? We are looking for the DNA which are released, extracellular. So what do? You take the supernatant and run the gel, the DNA gel, and see if you can find DNA. Or you could quantify the amount of DNA, eDNA they made. Number of ways you could assay that. Everybody clear? So these are part of the analytical tool you could use to monitor these different molecules produced by the biofilm forming cells. Okay? And that way you can assess the type of biofilm they are making, the quality of biofilm, the nature of the biofilm. So the function wise, you can see very similar to the other molecules: scaffolding, adhesion, cohesion, nutrient source, DNA damage, repair, gene transfer, interaction with other matrix components. Okay, so I think this is another point you have to remember. Remember, they are in the community of microbes, right? And if some DNA come out of those cells, so that means what? We are talking about their genetic material. Are you getting it? So the other microbes can take up that genetic material. Are you getting it? And so I'll talk about that later on, that the gene transfer can happen during this community or when they are in the biofilm environment. Because of this extracellular DNA, if it is a plasmid and it's carrying antibiotic resistance or carrying resistance to heavy metal or other antibiotic or or molecules or the resistance gene for different properties or different functions, and the other bacteria can easily grab it, right? And so extracellular DNA could be a source of transfer of genetic properties, right? And so that also another problem can happen within the biofilm community, within the cells among the biofilm community members. Okay, the lipids. Lipids, I think they don't produce that much of lipid. They are not known to produce microorganisms. I'm talking about. Okay, sometimes we are talking about, say, LPS, the phospholipid structure, right, or the cell membrane. That's a lipid structure, right? And so whatever the components or the amount of lipid these cells make, part of those lipid would be part of this biofilm matrix. Okay, so you'll find some of those, and as soon as you find them, so definitely they are going to be part of the components. So yesterday we talked about wild teichoic acid, right, and teichoic acid or wild teichoic acid, and that could be part of that. So they are not quite lipid, but with the association with the lipid bilayer, you'll be able to find it. The glycolipid, glycerolipid, sphingolipid, that's by candida albicans, and they are secreted, and they also have the similar type of functions. Lipopolysaccharide, these are some of those. That's the LPS. We talked about that. LPS is called endotoxin, and this endotoxin is associated with adhesion, colonization, host invasion, and also activate immune system very well. Okay, because this is endotoxin, and one of the function of endotoxin, again I mentioned that yesterday, it activates immune cells. Okay, especially the inflammatory cytokine. Inflammatory cytokines, say, for example, TNF alpha or IL-1, IL-6, IL-8. These are different types of inflammatory cytokines released by the immune cells. And one of the examples I was giving you, when there is any kind of infection, we get high fever, right? That's the first symptom we see. And the high fever happens because of these inflammatory cytokine levels. So, for example, TNF alpha. TNF alpha through prostaglandin pathway, it activates thermoregulatory center in the brain, and so the brain start to raise the body temperature. Why do they raise their body temperature? Because you go above 37. That's our body temperature, right? Centigrade we are talking about. So we get close to 39, 40, 41, 42, right? Sometimes that's serious, right? 103, 104, 105 degree Fahrenheit scale. And whole idea being is that the temperature can kill the organisms, right? That's the whole body's defense system. Body doesn't know. As soon as you have LPS or peptidoglycan, those sort of molecules in your system, body start to raise body temperature. And that's the sign of that, well, your immune cells are working, but if your temperature exceeds certain limit, what will happen? Why doctor tries to reduce your fever? High temperature is going to cook your body's internal organs, like Death Valley temperature. I was telling you, right? That joke. That's actually, it can internally damage your heart, lung, the brain, a lot of those organs, because they cannot withstand that high level of temperature. But the body's way of defense strategy, raising that body temperature, can kill those organisms which are producing these LPS or inducer, fever inducer. Getting it now? So that the organisms don't multiply or survive. But body didn't realize that raising that body temperature can destroy a lot of internal organs. And that's the whole idea. You need to reduce your temperature immediately. Whether take a shower, sometimes people are put into an ice bucket, ice bath, whole body submerged into the water to lower the body temperature. Okay, and whenever you have a fever, always go and take a cold shower. That's just the opposite or contrary to our practice, right? When you are fever, you just don't go close to the water. No, you need to take shower to cool down your body temperature. Okay, that's so important, because any duration of this high temperature can cause so much damage to your internal organs, you will never know. There is a lot of this other secret of this infection model is that a lot of times people die. It's not because of the organism killing you. It's your immune system becomes so hyperactive, and that also responsible for death. So, for example, COVID we had recently, right? So the COVID infection, COVID organism can recruit so much immune cells, or the immune system becomes so hyperactive, and they produce a lot of the cytokines at excessive level, which is not required, and their excessive immune response results in a lot of death or fatality. You heard about septic shock. Septic shock is the increased expression of a lot of those inflammatory cytokines in the body. And so certain molecules has to trigger it, right? So for example, anthrax. Anthrax is one of those deadly organisms, right? And people can die within 24 hours if you suffer from anthrax. There is another organism can kill you within 24 hours is Vibrio vulnificus. Vibrio vulnificus is one of those organisms. You'll find them in the water. Okay. Usually it's a foodborne pathogen. But if this organism get into your body through broken skin, so you have a cut skin and you are swimming in a water, there's a lot of Vibrio vulnificus in that water, and the organism can get through the cut wound, and then eventually that will multiply at a rapid rate, and this the cell outer membrane component LPS is gram negative, and that can stimulate immune system so rapidly, and the people can die of septic shock within 24 hours if it is not treated immediately. So you have to remember that. You heard about sudden infant death syndrome, SID? Anybody? You never heard about it. Sudden infant death syndrome. What it is that some infant, this is went to bed and next morning they are dead. Sudden infant death syndrome. Sudden infant death syndrome also is responsible by immune system, how it's aggravated immune response. So usually they predict, a lot of times we don't know exact cause of sudden infant death syndrome, is that especially the infants, sometimes they suffer from Clostridium perfringens infection, which is again foodborne pathogen, happen to be. Okay, Clostridium perfringens produce a lot of different types of toxins, and some of those toxins can activate immune systems to a level, and it's septic shock. Essentially, the infant can die very sudden, very rapidly, very quickly. Other organisms can cause that, Staphylococcus, which is again common organism, right? Staph aureus. Staph aureus makes tons of different toxins, and you may not have personally experienced it, but if you talk to certain people, which I have done a lot of this, and people talk about that, and when I teach, I bring that up, and this is not joke. I mean, people die, infant die suddenly. That's called infant sudden. That's what we call SID, SID. You can look it up in your microbiology book. Sudden infant death syndrome, SIDS, also they call it. Okay. So what I was trying to tell you is that when I'm talking in terms of this biofilm and this type of bacterial molecules or microbial components, and that is the reason I emphasized so much yesterday, talking about the bacterial cell structure, the fimbriae, the flagella, then the peptidoglycan, LPS, all of these different molecules, they play a tremendous role in disease process, okay, and especially the activation of immune system, which is critical. Okay, so as I mentioned you all those different types of molecules, and then again I'm going to show you this structure again. We published this as a review article 2021, and then whole idea is that during this biofilm formation, all of those molecules I talked about, how they play a role in forming biofilms. So look at this attachment. You are already familiar, right? When there is substrate, the bacteria are going to lodge, they are going to bind. Look at all those molecules I mentioned yesterday. Curly, I talked about curly, remember? Fimbriae, curly one type of molecule. Fimbriae, flagella, this is another pili or fimbriae. Teichoic acid, adhesion proteins, we just mentioned FNBP, BAP, SSG, SPA, ACT-A, LAP, some of these proteins. Depending on which organisms we are talking about, all these molecules help initiate that interaction or attachment, right? And all of these molecules helping forming biofilm, but if you look at them, they are all pathogenic factors. You see this? So the pathogenic factors playing an important role forming biofilm. And this is primarily for pathogens, right? Non-pathogens can have similar type of molecule, but they may not induce immune system. They may not cause tissue damage, okay? Now let's look at also microcolony formation. These are some of those genes or gene products are responsible for during microcolony. Maturation, you will find some of them. I already mentioned, we'll spend a lot of time about quorum sensing. And then these are some of those during this dispersal phase. Okay, and so whole idea being is that if you analyze closely, that each step of this biofilm formation are attributed to the production of certain gene products, right? The bacterial structure or microbial components, and that play an important role in biofilm formation. And at the same time, these molecules are important pathogenic factor. So if you look at the function, again, we are repeating few times that what is the function of this biofilm, right? So a lot of things happen. Can all of you read this slide from the back? Can we read it? Okay. So if you go one by one, extracellular polymeric substance, its role in the biofilm. Okay. One thing right here it says genetic exchange, right? So when the microorganisms inside the community, they're coming close to each other, right? And some are dying. I said they release their DNA, so there is a chance for genetic exchange. Whatever gene may be, whether it's a biofilm forming gene or antibiotic resistance gene or pathogenic factor, they can exchange some of those genes. Carbon source, polysaccharide, of course, that will be nutrient source, right? Pathogenicity or virulence factor, I already mentioned. They contribute to the pathogenesis. Biofilm structure, right? Then also there's a symbiosis with plants. Sometimes this carbohydrate helps them to have a symbiotic lifestyle with the plant material, okay? Because microbes contributing, plant contributing, both benefits, okay? Not necessarily just for pathogens, okay? You can rule out which are important for pathogen, which are for any generic organisms. Drought protection. So if there is a drought, no moisture, water, this microorganism kind of encapsulated inside this structure, they hold the water, right? And so that can prevent or protect from drought. Aggregation, adhesion, trap for nutrients, obviously it's trapping a lot of nutrients. Also a lot of signaling molecules, we'll talk about that, quorum sensing and signaling molecules. And protection from antibiotics, we've been talking about that, right? Antibiotics can be absorbed in that or prevent antibiotics from getting to the individual organism, okay? So there's a lot of functions. Then again repetition kind of. So if you look at the functions of the EPS, adhesion, aggregation, cohesion of biofilms, retention of water, protective barrier, sorption of organic or inorganic compounds, enzymatic activity, nutrient source, genetic exchange. Again, you have this source, make sure to read it. You will learn very well the biofilm is multifaceted, okay, and it contributes so many different functions. The benefits of biofilms we already talked. Microbes held together by exopolymeric substance. We already talked. Protection, shelter for pathogens, nutrient source, persistence or persister cells. We'll talk a little bit more about that. Antibiotic resistance, low metabolic activity. Obviously, cells in the sessile stage, their metabolic activity is low. They're kind of like a dormant stage or called persister phase. They are not taking a lot of stuff. They are not taking food. They are multiplying, dividing. And so if antibiotic needs active invasion or active uptake, this antibiotic is not going to be absorbed. Therefore, those cells become resistant. Okay, so that's one of the mechanisms. And of course, in the pathogenesis is very important. Adhesion, attachment, also immune evasion. Biofilms and diseases. Again, you have seen this slide before. We talked about different types of diseases where these biofilms are very important. And almost you can see this: Staph mutans, Staph aureus in mastitis, intestinal colonization for regular infective diseases, endocarditis, the heart valve problem, cystic fibrosis, lung infection. Okay. So virulence factor involved in biofilm formation, already I mentioned that to you, but if you look at it again, these are all these pathogenic factors contribute to biofilm formation. So it's kind of beautiful for microbes, right? So if I have a one molecule, I can do two types of function. You see this kind of redundant build system is that I have virulence factor. When I'm infecting a host cell, I can use that protein to bind, right? And I can invade. But at the same time, if I need to make biofilm for longer survival, I can use the same protein again. And so that's the beauty of it. You need to start thinking, not in isolation, think in combination, the redundant function. And so the microorganisms use the same protein for multiple functions. Okay. Um, and then this one, virulence factor involved in biofilm formation. Again, what I just said before, a lot of these proteins are involved in pathogenesis during infection. Okay, and also they contribute to biofilm formation, and then you can see this pathogenesis. This is Staph aureus. So we again, this is from that review article I mentioned. It's listed these tables. So if you are interested on certain organism, what are those proteins or the factors that contribute both biofilm formation and pathogenesis, you can get that from this review article. This is more again for Salmonella, E. coli, Pseudomonas, the different types of proteins, what's their biofilm function and their function in pathogenesis. We really clear the summary of it, the dual role of a lot of these protein molecules produced by microorganisms. Okay, so summarize this part of the work. Biofilms are produced by bacteria, yeast, mold, parasites, and then often they make polymicrobial biofilms, right? Structure is pretty generic, but sometimes unique depending on which organism you are looking at it. Very generic, right? They will come and attach, form microcolony, then they will form mature biofilm, then dispersal or detachment. That's when the sessile cells come out. So that steps you should never ever forget, right? Only thing you want to remember how these biofilms are formed, right? So that's very generic. Regulation, of course, a lot of functions we talked about, and contribution to pathogenesis. That's very important, right? Okay, so that's the end of this chapter. Any questions before I go on to the next lecture, which is supposed to be what 2 o'clock or 3 o'clock, but I'm going to start that now because we have some time. What?
In laboratory, when we isolate DNA, we used to stabilize it with TAE or TBE, store it.
Okay.
But in EPS,
It's the same thing. You can do the same treatment to assay it.
but naturally in a biofilm, why it won't disintegrate or lose its identity?
Okay, I would publish that work. There is a method you can use it to differentiate from eDNA from the actual DNA. That's the question you were asking.
No, I'm asking that in biofilm, if eDNA is present
Okay.
in the EPS,
Okay.
why it won't lose its identity? Like it remains as eDNA outside the cell.
Yeah. Yeah, it will be there in that matrix as a extracellular DNA. That's the question, right? And yeah, I mean, it is going to be there. You can separate it. So the way we try to assay it, if they are producing eDNA, I don't know. I'm trying to understand your question, or is there a question built in?
I think she's saying that the eDNA, how it is stable in that matrix.
Oh, how it remains stable? Well, DNA are generally very stable, right? I don't know. The matrix itself, you are worried about the nuclease enzyme produced by organisms or others. That can damage it, but still finding DNA outside the cell is kind of unreal, right? DNA always inside. You have to lyse the cells to get it out. But when you analyze the biofilm and finding DNA outside, whether it's a broken or you are not going to find an intact DNA. No, I don't think you're going to find intact DNA like if you get it from the cell. So it's like whether it's a broken or chopped or cut, degraded partially, but still the DNA is DNA, right? It's going to be there. So your goal would be, if it is the eDNA contributing to this biofilm formation, and you should be able to collect that matrix or molecule and run in the agar gel with the ethidium bromide staining. You show that, well, or cytogreen or whatever you have, and you can show that yes, there is a presence of DNA. And as soon as you see that, that means it's one of the characteristics of the biofilm. Oh yeah, I'm not going to ask for the stability. It may not be stable in that environment. because a lot of enzymes, nuclease being released by others. Yeah, that answers the question. Okay. Any other questions?
Sir, am I audible?
Yeah. Yes.
Sir, my question is also related to eDNA. Like you mentioned that eDNA is involved in genetic transfer in the EPS. So, sir, there are many biofilms with MDR strains residing in it. So, this gene transfer due to eDNA, might it be possible that this gene transfer specifically causes these strains to become MDR or multi-drug resistant?
Drug resistance, that can happen, and that's one of the concerns, that within that microbial community or biofilm, and the DNA released by one, say salmonella, multi-drug resistant genes, right? The genes can be taken up by other microbes within that community, and they become resistant. And so, that's a great possibility. And so, speaking of that biofilm and genetic transfer, also other place it can happen, right? If you think about in our gut, we got so many microbes, microbial community in one location, and that really a big possibility. A lot of times we see the evolution of new organism coming up. A lot of times they are in this community in a small room, small compartment, and they are all growing and competing. A lot of times, a lot of the genetic material also transfer, like biofilm, also in this community when they are in close proximity, and that can happen. And it doesn't happen at high frequency, then that will be a big problem for human community. But it happens nonetheless, even at low frequency. There's a possibility it can happen more. Okay.
Thank you, sir.
Yeah. Any other question?
Sir,
Go ahead.
my question is related to the small portion that you mentioned regarding the flagella turn-on gene.
Uh-huh.
So, can this be explored for controlling biofilm? So, you said that flagella gene is turned off during the biofilm formation.
Yeah.
So, if it is continuously turned on, so can the biofilm formation be inhibited in that way?
Well, that's a good point, though. But it's kind of like a natural process for the microbes. So, like, I don't need, for example, so that's a good point. When microorganisms invade a host cell, so sometimes they use flagella to get to that site. But then, if they once they are inside the host cell, do they still express flagella? So the question is no, they don't need, because they say I have no need for flagella when I'm inside the cell, which is not going to help me to go inside. And once they are inside, they would focus on upregulating genes that will help them survive better inside the host cell compartment. Okay, so I need certain energy, need some enzyme to harvest from the host cell. So I will also produce a lot of enzymes, and so that I can get substrate or the product from that host substrate. So that's what they do. So the question you were asking, the parallel is that when they're in the biofilm, generally they turn it off. Okay, and I think I was trying to show you one cartoon before, that flagella are on when they are under normal conditions, and when they are under stress conditions, they said I don't need the flagella anymore because I am inside this the biofilm cell matrix. There is no need for flagella. And so you are saying that if you keep on expressing, probably when they are trapped within the matrix, I don't know whether they will be able to get out of that. And I don't know whether you can force them to say genetically manipulate, turn it on, right? Probably that will be a very difficult task under that conditions, the environment you are in. Okay, they can keep on producing, but it's not going to be any use. So therefore, there is no benefit. Okay, how do I get out of this? I think I pressed the wrong button. What is that? Yeah, go ahead. Go ahead, start. Okay, can you get to my slide? Get out of that.
Sir,
Yes.
so my question is, eDNA release is active or it undergoes by the process of autolysis, and how much stable it is in under environmental stress?
Yeah, that was the same question she asked us. So I don't expect that to be a real stable intact DNA. So under biofilm conditions, I'm suspecting that a lot of enzymes or nuclease are produced by other microbes in that environment. So I don't think that they are going to be stable. That's not the point. The point here is that when the DNA is out, it makes a kind of sticky condition, sticky environment, and that stabilizes the biofilm structure. And so stability is not necessarily an important question to ask at that conditions, whether they remain stable or not. If you think about carbohydrate or protein, all of those can break down into small fragments, right? And as soon as the nuclease breaks down the DNA, so that can be a source of carbon too. Bacteria, microbes can use that as a carbon source, energy source. And so stability not necessarily an important consideration to think of when they are in the biofilm. But nonetheless, they come out, they contribute to that, but at least we can recognize two functions. It helps forming the biofilm, right? And second is that the genetic material it released, other microbes can pick it up, and they can integrate within their system, and then they become resistant, or they can inherit that gene, and that can propagate the pathogenicity or whatever function may be. Can I answer your question? Okay. That's a very good question. Any other question? Yeah, let's get that now. I know we are almost. We can. Okay, so not start this now. That's what you're suggesting. Okay. Any other questions, so that we can take advantage of it. 5 minutes or so we have. Ask the. Can you repeat that question?
How can you differentiate the eDNA and the cellular DNA?
Oh, okay. Is there a way to differentiate, or how do you differentiate? Well, so the way you assay it, there are some molecular techniques. Right now, I cannot tell you specifics, but give you general answer. So, if the cells release the DNA, right, so that will be outside, and if it is intracellular, it's inside the cell. So that means the cells which are dying, they release that, right? And the cells which are intact or living, usually they are inside. And so the way we do our assay, we have done that. I'll share some data this afternoon, and is that you will allow the cell bacteria to form biofilm, right? And if they are secreting eDNA during this biofilm formation, then what we do, we take the supernatant and run the gel. Okay, and usually you should not find any DNA, but if there is a eDNA, then when you run the gel, you'll be able to see it. And then if you lyse the cells, then you will see even more, right? But that one after lysis. But this is just the free DNA, you'll find it in the supernatant. And then there is another way you can do it. A lot of times you can use certain dye. So for example, ethidium bromide, right? And so if you treat the biofilm with the ethidium bromide, and ethidium bromide is going to intercalate within the double-stranded DNA, right? And it's going to intercalate only in the eDNA because it is outside, right? But the cells which are intact, but their DNA is inside, ethidium bromide is not going to get in, right? And so now if you extract the DNA and from the biofilm and from the intact cells, and you run the gel, and you can see that the DNA which is outside, or eDNA, you'll see they are stained. The other one will not stay stained. So a lot of different ways you can assay it. But those are really important because I'll show you some data, even though I'm not going to talk about the actual experiment, but I will show you that this bacteria forming biofilm and they're producing eDNA, you can clearly see it in the gel.
Thank you, sir.
Okay.
biology student from Vishwakarma University. I think it's better that we have to ask them. We should ask questions.
Okay, we'll ask them the questions. Yeah, so this is the most important part of the biofilm structure, right? We spent this lecture.
Good morning, sir.
Yeah.
My question is, can we use chelating agents for controlling biofilms?
Chelating agent.
Yes.
Yes, I think you can use. I think we'll see that controlling biofilms using chelating agents. Yes, I think you can use it. Depending on, there are different types of molecules.
Like, can we design any assay, or it is already existed, like EDTA based or chelating agent based assay to detect biofilm or to control them?
Yes, I'm going to talk about that when I think it's towards the end, control and prevention of biofilm. There are a lot of strategies we'll talk about that. But the answer is yes. You can use chelating agents, a certain surface modification with the chelating agents. You will be able to control biofilm formation.
Oh, thank you, sir.
Oh, there's a break.
Streptococcus and its association with antibiotic resistance. So, like from the starting onwards, we have been discussing that the biofilms also results in emergence of antibiotic resistant pathogens. So, she has correlated this antibiotic resistance with the biofilm formation in group B Streptococci species. So, welcome Dr. Pooja. So, over to you.
Hello. Thank you, sir. Can you hear me, guys? Okay. So, welcome you all. Today I am going to discuss about the biofilm in a pathogen which is group B Streptococci. So I hope till now you all are aware what are the biofilm, how do they are being formed, and what are the advantages and disadvantages of these biofilm. But today I am going to be more specific about the role of these biofilm in a bacteria, and more specifically in the bacterial pathogens which causes disease in the humans. Okay. So, just going to brief about the biofilms. So, when we talk about the biofilms, what are the biofilms? These are the community of bacteria. So community like same as the humans community, we live in a community. So these bacterial community are protected by a matrix which is produced by the bacteria. So these are the self-produced polymeric matrix and biofilm are attached to the surface. So this is just a general picture of the biofilm here, and you can see the bacterial colonies here attached to the surface, and you can see the differentiation in the color. Some are red, some are yellow, some are oranges. Why is it so? Because you see in the core of the biofilm, these are the bacteria which has a less nutrient, which has a access to the less nutrient or oxygen, and these bacteria, they have a more access to the nutrient and the oxygen. So this biofilm, the matrix here, the yellow one, it is composed with the polysaccharide proteins and the nucleic acid. You can see the nucleic acid and the polysaccharides and the protein here. So if you want to define it, it is bacteria adhere to each other. You can see they are adhering to each other and the surfaces here, embedded in a sticky protective matrix. The formation of the biofilm enables bacteria to survive hostile environment, resist antibiotics and evade the immune system, which is a very much specific about the pathogens, because we are talking about the pathogens. So how do the pathogens escape the human system? Because you all are aware that human, they do have a immune system. So once the bacteria, they are in the biofilm, they can evade host immune system, they can persist there, they can develop the resistance toward the antibiotics. So nowadays you heard about the multi-drug resistance, isn't it? So how this multi-drug resistance achieved through the formation of the biofilm, we will discuss those things in a later slides. So the matrix here, this contains water, nutrient and signaling molecules like quorum sensing molecules. So what is quorum sensing? So I told you bacteria, they live in a community, okay? We humans, we live in a community, we talk to each other, we go to each other places, we greet each other. How bacteria do that? So quorum sensing is like a talking. Bacteria talk to each other through these chemical substances, and this whole network is known as the quorum sensing, okay? So whenever you heard about the quorum sensing, that means it is a bacterial network or the way bacteria talking to each other. Surfaces, so these biofilms, they can be formed on the teeth like a Streptococcus mutans. It can make the dental plaque and the human tissues and sometimes on the catheters. So you may heard about the hospital acquired infections. So how these infections get transmitted, because the biofilm can be formed on these devices like a catheter and all these things, and when they are used in the patients, they can cause the disease. So they provide stability, enhances bacterial survival and increases resistance. So microcolonies of a mature biofilm are typically characterized by the presence of an EPS matrix that is composed of polysaccharide, DNA, protein, lipids and enzymes. So now bacterial biofilm, it is, we already discussed about it. So it is made up of this matrix which contains 65% of this biofilm matrix, and 35% is the bacterial population. Biofilm architecture contains densely packed microbial cells, and it has a water channels as well as polymorphic structure adjusts based on nutrient availability. So if they have a, like here I put an example like a glucose as a nutrient. So if they have a high glucose concentration, means high nutrient concentration, they make a faster microcolony, they can multiply more rapidly, and that's how they make the thick biofilm. In contrast, when the nutrients availability is low, then there is a reduced biomass and the biofilm that is produced, it will be thin. Then composition of the EPS. So EPS is 65% of the biofilm, and this 65% of this matrix, it's most of that is the water. 97% is the water. 1 to 2% is your polysaccharides, then protein and DNA, RNA. So these all these materials, they are secreted by the bacteria. So this is just a life cycle of the biofilm. So what happens when bacteria, they find a surface to which they want to bind or to adhere through the adhesins. Okay, so adhesins are the surface proteins, or here you can see this fimbriae or sometimes small hair-like structure that are known as the pillae. So through these adhesins, bacteria can attach to the surface, any surface. So here they are just being attached, or we can say that they go to in the colonization phase. Once they found a suitable condition, then they go in this phase and they start multiplying, and during this stage they start producing biomatrix, biofilm, biomatrix. Once they have all the favorable condition, they make this fully fledged biofilm, and there is a stage when these biofilms get mature and then they get dispersed. Okay. So what happens when biofilm get dispersed? Now this bacteria, so it will be in a which stage now? It will be in a planktonic stage. So planktonic means free floating, and biofilm or sessile means attached to something. Okay. So when biofilm get dispersed, now these planktonic bacteria can go and bind to any other surface, and then again they can produce the biofilm. Okay. So biofilms are highly adaptable to environmental changes. They communicate by chemical signals that we know as the quorum sensing, produced and interpreted by other cells in the community, both of the same and different species. Okay, here I put different species. So if I talk about the dental biofilm, what happens? It's not a single bacteria. Several bacteria are together in this biofilm. Okay, so it can be a same species in the biofilm, or there could be several species in the biofilm. So quorum sensing, mediated through the chemical signals, it allows bacteria to talk and listen. So how would I pronounce it? Like a bacterial language. Okay. So they talk and listen to each other, triggering population-wide behavioral and genetic changes. I am not going into detail, but what happens that during the biofilm formation, bacteria, they talk to each other and they can regulate the expression of several virulence genes. Okay. Then this coordinated biofilm behavior enables survival against the host immune system and antimicrobial treatment. So this is just another diagram. Here the biofilms, they are in the planktonic stage. Here you can see the bacteria, and now they are attached to the surface. Now they are in the sessile stage, and then multiplication takes place in the bacteria, and then later on the dispersal. So this is the process of the biofilm formation. So this is the initial attachment, free floating bacteria are here. Then this mechanism is the reversible. Okay. Reversible means they can be detached in the first stage. Later on, this stage is irreversible. Means once it is formed, it cannot go back. Okay. So this is the irreversible mechanism. Then microcolony being formed, and then maturation of the biofilm, and then dispersion of the biofilm. So I hope it is clear to you that how biofilms are being formed. So just a few examples here. So I told you that bacteria go and attach to the surface. Okay, that is the first step. What happens in context of the bacterial pathogen, if they do see some favorable conditions during this attachment, there will be more chances of forming the biofilm compared to the other conditions. So here I put an example of Pseudomonas. Pseudomonas aeruginosa. If it found a surface which is coated with the mucin, it will make more pronounced biofilm compared to the non-treated or uncoated surface. So the reason behind telling this, that the treating or conditioning of the surface makes a huge impact on the biofilm formation. So this is the reversible attachment, as I told you, that lead to irreversible attachment, then colonization and detachment. Now coming up to the main question, how biofilm contribute to the antibiotic resistance. Okay, it is the whole complex network actually. It's a whole molecular network, and I am discussing very few on the superficial level only. Okay, so the first step is the mutation. Okay, so bacteria in biofilms are more resistant to antibiotic, like for example aminoglycosides, fluoroquinolones and beta-lactamase than the planktonic, because they have some mutations which allow them to attach to the surface and then inhibiting the antibiotic. Okay, that is the one condition. And how it happens, just giving you example, I think you might aware about it. So beta-lactam antibiotic bacteria, they do have a mutation, and they beta-lactamase, they produce beta-lactamase, and the antibiotic can no longer work on these bacteria. So that is due to the mutation in the PBP gene. Then some bacteria, they do have a resistant phenotype. They contain special groups of bacteria that actively resist antibiotics. The third is stress and resistance. When bacteria and biofilm, they face any stress like a lack of nutrient or crowding. Crowding means like when multiplication is being happen in the bacteria, lot of bacteria, they are getting multiplied. Okay, so that is also a type of a stress in the bacterial community. And then antibiotics. So these stresses, when bacteria face them, they change their membrane proteins. This makes it harder for antibiotics to enter the cell. The next is nutrient and oxygen gradient. As I told you, there is a limitations of the nutrient, isn't it? When bacteria are actively dividing, they have a lot of nutrient, but at some point, nutrient get depleted. Okay, so when it happens, the biofilm surface, they do have oxygen and nutrient are available, but deep inside conditions are low in oxygen and nutrients. This slows bacterial growth, making them less affected by antibiotics. And during this condition, some bacterial cells turn into persister. There is a terminology known as the persister. These are metabolically inactive bacterial cells, and these persister can go back to the normal bacteria, and that's how they develop the antibiotic resistance. So you might heard about, like, you take the antibiotic, okay, infection is gone, but after some time it comes back. It comes back due to the persister cells. Okay. Then adaptation to hard conditions. Biofilm bacteria can survive stress like low oxygen, high temperature by activating protective genes and producing enzymes that break down antibiotics. Then quorum sensing, chemical talk. These chemical signals, their communication triggers the production of toxins and enzymes that protect them from the antibiotics and the immune system. And the last one is the efflux pumps. Efflux pumps are the one when antibiotics, so these all here till 6 point, antibiotic is not able to enter inside the bacterial cell. But somehow if it get inside the bacterial cells, then efflux pump work. What they do, they pump out the antibiotic outside the bacteria. Okay? So in that case, antibiotic cannot work anymore on the bacterial cell. So biofilm, it provides the resistance against the antibiotics, as well as it can regulate the expression of several virulence genes. So the question comes, how biofilm contribute to antibiotic resistance? So as I told you, the penetration of antibiotics, the antibiotic cannot go and enter the bacteria. Due to the biofilm, they have this sticky layer called the glycocalyx or exopolysaccharide. This layer protects bacteria inside the biofilm, or they can block the antibiotic, like the matrix, it will stop the antibiotic and it slows down how fast antibiotic move through the biofilm. Like example is ciprofloxacin, it binds to parts of the biofilm which reduces its ability to reach bacteria. And how it works? So bacteria, its cell wall is what is made up of? Any idea? Yeah, and it is negatively charged or positively charged? Negatively charged. Okay. So these antibiotic, when they come to these polysaccharides, they have a interaction with the polysaccharides, and then they bind to the polysaccharides, or I would say they get hindered, or they get blocked inside the polysaccharide, and they cannot enter into the negatively charged bacterial cell wall. Other antibiotics, like antibiotic stick to the biofilm, they don't move through it well. Aminoglycoside antibiotics, because they are positively charged, it binds to negatively charged polymers in the biofilm matrix. Okay, Because biofilm matrix is negatively charged, they are positively charged, interaction will happen, and then it won't be able to interact with the bacteria. So now that was the mechanism I told you about the how biofilm can contribute to the antibiotic resistance. Now we will discuss one by one some of the species of the streptococci, and in these streptococci how these biofilm are producing and how they are similar to other streptococci or differ from other streptococci. So I just put 5 or 6 examples of different streptococci species. The first one is Streptococcus pyogen. So Streptococcus pyogen, or we call is a group is streptococci. It is a flesh-eating bacteria. So if you get infected with this group is streptococci, suppose you get prick in your finger, then you have to cut your finger. Okay. Otherwise, group is streptococci infection can lead to other organs, and then it will lead to toxic shock syndrome. So what happens when we talk about group streptococci biofilm? Researchers have found some few very specific things which happens only in group streptococci biofilm. So they have glucose and mannose, which are the major sugar in biofilm matrix, and they also found, so this is flagella and pili. In addition to that, there are microbial surfaces component recognizing adhesive matrix molecule. This is a terminology for that. So they have several proteins which act as adhesins, and through this pili protein or these adhesins, bacteria go and bind to the surfaces. More interestingly, there are more than 100 serotypes of the group A streptococci, and what researchers have found that each serotype of a streptococci, their biofilm is very specific. For example, some group A streptococci serotype, they don't make any biofilm, whereas others, they make a lot of biofilm. Okay? So we can say it is strain specific characteristic and virulence. For example, there are some serotypes, you see AM6 and 14, they make a strong biofilm on abiotic surfaces, whereas 49, 12 and 1, they do not make any biofilm. Okay? So now this research is going on that how few serotypes are producing more, few producing less, what is the connection between these, are they more pathogenic, are they less pathogenic, and these sort of the things. So species specific targets, AM proteins, pili, and then other adhesins, they involved in adhesin in biofilm matrix. So how can be biofilm mediated infection can be treated in group A streptococci? So use of anti-M protein antibodies or peptides and inhibitors of pilus assembly. So if you use inhibitors of pilus, pili won't be able to interact with the surface. If there is no attachment, then no biofilm formation. Okay. The next one is beta hemolytic group C and G streptococci, more specifically Streptococcus dysgalacti, subspecies Equisimilis, SDSE. So it causes biofilm formation in SDSE varies with EMM type. Same with the group Streptococcus, there are different EMM type, and they have found that biofilm formation varies in different EMM type. So here biofilm is being formed, then extra eDNA has been implicated in biofilm formation. So biofilm matrix has a protein, it has a polysaccharide, it has a eDNA. In case of this bacteria, they have found that eDNA is more prominent in making the biofilm. So SDSE biofilm formation enhances resistance to host defenses and antibiotic, contributing to persistent infection. It is strain specific, and as they have seen, some bacteria, they produce strong biofilm, whereas others produce weak biofilm. Okay. The other example is Streptococcus pneumoniae. We all know it is colonizes and human nasopharynx. It is a major pathogen, and it infects the respiratory tract. And biofilms have been detected in clinical settings such as pneumonia, otitis media, and rhinosinusitis. So clinical implications that biofilm being formed in pneumonia. So before that, Streptococcus pneumoniae has a more than 100 serotypes again. Okay. So biofilm formation is inversely correlated with capsule formation, which means if there is a lot of capsule being formed, biofilm will be reduced. Okay, so it is inversible correlation. Highlight the presence of acetyl glucosamine and beta-linked glycopyrenosyl units in pneumonia biofilm. Extracellular DNA and pili protein. Again, pili are a major contributor. GA, GB and C, these are the major component and they play a role in the biofilm formation. And how did they came up with this theory? When they make the mutant, suppose they made a mutant in RRGA, they found there is a less biofilm formation, and that's how they concluded that these adhesins playing a role in the biofilm formation. Then dispersion, it is involved in the development of invasive infection. So invasive means when bacteria get inside the cell. Okay, that is the invasive infection. Colonizing means bacteria just colonizing the surface, do not penetrate the cell. So I am not going to discuss this whole cascade of signaling in Streptococcus pneumoniae. And another one is mutants. So mutants causes the dental plaque. So if you have a tooth decay, this is the main corrupt bacteria here. So here is the biofilm, and then there are several proteins like a glucan binding protein. They stabilize plaque on tooth surfaces and then interact with biofilm matrix. GFTs, these are the genes that produce a key component of the biofilm and produces glucans. So these are GFTB and GFTC. They synthesize water insoluble. Some synthesize water soluble glucans. And then another one is antigen C, that bacteria cell surface protein antigen. They mediate adherence of bacterial cells to tooth surfaces and via salivary follicle. This one example is the where several other bacterial species were also involved in the formation of the tooth decay, as mutans, as angionosis and others. So as mutants forms biofilms on tooth surfaces contributing to dental caries. Involved include glucan binding proteins, collagen binding proteins, glucosyl transferases and antigen C. Now coming up with the one GBS that I will go in detail and discuss those one. So I am just skipping it. Just invasive, there are 10 serotypes of group B streptococci. Some are invasive, some are colonizing. Okay. So the biofilm is being formed here. So there is a study which has found that there is a higher or a strong biofilm is being produced in the colonizing strain, not in the invasive strain, and it is very interesting to know, because accordingly invasive are more virulent, isn't it? On the acidic conditions, like the pH is very low there, so acidic condition they helps the bacteria to form the biofilm over there. It facilitates colonization and survival in the host, and more importantly they have found that protein is very important in biofilm, because when they treat the biofilm with the proteinase K, the biofilms diminishes. Okay. So clinical implications and strain specific characteristics. So I am not going to discuss in detail. If you want, I can share with these slides with you. It's just a summarize of those similarities between the biofilms of all the streptococcal species that I discussed earlier. So common target across streptococci, surface proteins and pili, extracellular polysaccharide, quorum sensing system, and then extracellular DNA. So how the biofilm is regulated, okay, in streptococci. So like a quorum sensing, CSP pathway in Streptococcus pneumoniae, mutants and other streptococci. So mechanism, CSP is a small peptide which is secreted and accumulates as cell density increases. When a threshold is reached, CSP bind to COMD receptor, activating the COME regulator, which triggers the expression of competence gene and promote eDNA releases, contributing to the biofilm matrix. So species, I am not going to discuss it. Then there is a two component system, Lux S and AI2. AI is an autoinducer 2 which is produced by the Lux 2, and they all, this gene cascade play an important role in the biofilm formation. Then short peptide based system, it includes RGG and SHP and SIL system, and these hydrophobic peptides interact with regulators to modulate biofilm gene expression. Okay. So what is the significance of biofilm formation in group B streptococci? It enhances colonization and persistence because it has been seen that colonizing strains have a more strong biofilm compared to the invasive strains. They have a role in the virulence because the biofilm is strong biofilm producing bacteria and then less biofilm producing bacteria. When they did the comparative proteomics or they did the other techniques, they found that there are a specific set of genes or proteins which were involved only in biofilm formation. Then clinical implications, they are involved in sepsis, meningitis, pneumonia, bone and joint infection. Biofilm confer antibiotic resistance to GBS. We will discuss in detail. We can skip this one. Okay, it's very interesting to know. So in GBS, the two component system, so it's a genetic regulation. It regulates the whole cascade of the virulence gene involved in it. So SAERS, it promotes biofilm formation. Here you can see they are making more biofilm, and when they knock out, okay, when they delete this gene SAERS, they found it is making no biofilm compared to COV-RS, which is another two component system. When it is present, bacteria make less biofilm. When they took out this gene, they found that it is making more biofilm. Then environmental regulation, if they have a more nutrient rich conditions, they have robust biofilm. Extracellular matrix component like human fibrinogen, they strongly helps GBS to bind to the fibrinogen and then promote the biofilm formation. Okay, coming up to the, so that whole part was review part. Now I am coming to the research part, and I have only 30 minutes, so I will go quickly with that. So the biofilm formation and antibiotic susceptibility of group B streptococci isolated from Indian population. So before that, there is a now like a huge thing is going on about the antibiotic resistance. It increasing. Lots of cases of not only the MDR, now XDR is being reported. So it is estimated by, so this is the data of 2020, the pink one. There is only 1.3 million cases of AMR, but it predicted by 2050 there will be 8.7 million. So you can see that how antibiotic resistance is going to be like a huge burden on the human population. So now we are looking for the alternative to treat the antibiotic resistance. So the introduction about GBS, it's a gram-positive, beta hemolytic, it's a human pathogen, there are 10 serotypes. So what happens when, so I told you it is in the female population. Okay, so when women are the pregnant and its bacteria is residing there causing no infection at all. When neonate or newborns, they are being delivered, the bacteria somehow reaches into the newborn, it gets into their brain or in their lungs, causes meningitis and pneumonia, which lead to the death. Okay? Even in immunocompromised adults, group B streptococci can disease these life-threatening infections. So, to treat the infection, penicillin shots are given to the female. So it is very common practice in United States. If a female is pregnant, it's mandatory for them during their third trimester to go and check for the group B streptococci. If they found positive, then the penicillin shots are given to those females. But this is not in India. Even we don't know what is happening in India. What is like group B streptococci? What is the ratio or what is the actual number of bacteria causing disease here? Okay. So the connection between biofilm and antibiotic resistance is of considerable interest. GBS can produce biofilm, so unlike other bacteria. So this is the clinical manifestation. It causes different diseases in the humans. This is the worldwide distribution. In India, we have no cases of group B streptococci. So it doesn't mean that we don't have cases. The problem is no one look into the what is actual infection B caused by the group B streptococci. So no cases reported means they says no cases are here. So what I did during my PhD, we collected the samples from several patients and we found that there is a huge burden of group B streptococci in Indian population, which need to be addressed actually. Okay. So we published published this data and we did a serotyping. As I told you, there are the 10 serotypes of this bacteria. We did the serotyping, then we wanted to know that which one is colonizing, which one is invading, which is virulent, which is less virulent. So that was the work has already been done like 15 years back. So now in my lab, we wanted to know what is the antibiotic susceptibility pattern of group B streptococci. Are they resistant? Are they susceptible? What is happening here? Then we wanted to know, do they make biofilm? If they make biofilm, it is connected to the antibiotic resistance. And then the association. So this work was done by my PhD student Shalini. So prior to this study, there was limited understanding of the association between antibiotic resistance, virulence genes and biofilm formation in GBS strains. So what we did, first to understand the antibiotic resistance by different GBS isolates, we used different antibiotics, okay? And then we look at the zone of inhibition to check their resistance, whether they are resistant or susceptible to these selected antibiotics. So interestingly, we found here you can see that 27% of GBS isolates are resistant to penicillin, which is quite interesting because till now only in Japan or in one more country they have reported penicillin resistant group B streptococci, that's it. So that was a major concern. If group B streptococci getting resistant to penicillin, what would happen? Because I told you in USA they are giving penicillin shots to pregnant women. But if they are resistant, what would be the alternative mechanism to treat this resistance infection? And we use other antibiotics as well. So here we show there is a lot of like a 30% of isolates they produce the penicillin resistance. Then we wanted to know what is the association between the GBS serotype and antibiotic resistance. So we looked at the specific serotype because we had a collection of type 1A, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay, so these are the 10 serotypes. So we wanted to know, okay, if serotype 1A, is it more antibiotic resistance or serotype 3 is more antibiotic resistance? So we have a different isolate. Suppose we have a 10 bacteria from serotype 3, 2 bacteria from serotype 1A and so on. So we look at the antibiotic susceptibility pattern of all these isolates and we wanted to make any correlation between the serotype. But we did not find any correlation, so it is not serotype specific. Then we look at the biofilm, whether they make a biofilm or not, and for that we use two assays, qualitative and quantitative assay. So the one is the Congo red agar assay, which is the qualitative assay, which that just let you know, okay, it is making biofilm, that's it, yes or no. Another one is the crystal violet assay, quantify method, where you can quantify how much biofilm is being produced by the bacteria. So this is the Congo red agar assay. How this works, the dye Congo red, it binds to the proteins and polysaccharides present in the matrix. Okay, so more biofilm, more dye will bind, isn't it? So you will get a red, dark red or brownish color, okay, because dye is binding to the biofilm. If dye is not binding to the biofilm, means there is no biofilm, so you will get something like this, okay. Then other method is a crystal violet assay, where you stain your bacteria with the crystal violet. It works in the same way. Crystal violet is a dye which go and bind to the cell wall of the bacteria as well as polysaccharides and proteins of the biofilm. So more biofilm, more bacteria means more binding of the CV assay, and then you will get more color. So here you can see this is our positive control. These are our group B streptococci, and we used a control E. coli which does not produce any biofilm. So what we found that almost 70% of group B streptococci, they make a strong biofilm, 30% make a moderate biofilm, but we did not find any GBS isolate which do not make any biofilm. Okay. Then we did the crystal violet assay. We wanted to know which serotype is making more biofilm and which is making less biofilm. And we found a connection here. We found that serotype 24 here, it is making more biofilm, and G7 it is making the less biofilm. So then we wanted to know what is the association between antibiotic resistance and biofilm formation. So let's put it in this way. We have 30% of GBS isolate, they are penicillin resistance. Okay, we have a GBS isolates which are making a strong biofilm and moderate biofilm. Then we looked at penicillin resistance GBS serotype and their biofilm forming capability. And it was so interesting to know only those GBS isolates which are making a strong biofilm, they were penicillin resistance. Okay, which proved the hypothesis that biofilm can lead to the antibiotic resistance. So we used some statistical analysis and we found a correlation that whenever penicillin resistance bacteria are there, they are making the more biofilm. So the another case we wanted to know, as I told you earlier, that biofilm through the quorum sensing, it can regulate the gene expression, isn't it? So now we wanted to know, we have a strong biofilm, we have a moderate biofilm. Now we wanted to know, are these strong biofilm, do they have some like a different kind of virulence gene, or the expression is different compared to the moderate? So we chose a few genes which were earlier reported in group B streptococci. Those are involved in the virulence. Okay, and what we wanted to know, whether the presence of some specific virulence gene is directly correlated with the biofilm. Okay, as we found in case of penicillin. So we screened virulence gene by the PCR. Okay, when we ran these PCR product on the gel, and we wanted to know the presence or absence of the biofilm. So here you can see these are the strong biofilm, these are the moderate biofilm. No association. Don't you think it's like a random? Okay, so we came to the conclusion, it doesn't matter whether there is strong biofilm or moderate biofilm, there is no association with a specific set of the virulence gene. So the conclusion we can say, high frequencies of the virulence gene, they were detected in GB isolates, and most isolates carried multiple virulence gene, which allowed the GBS strains to adhere, colonize and invade the host tissue. However, there were no association between virulence gene and the strong biofilm producers. So we will come to the conclusion. We reported occurrence of 30% penicillin resistance followed by erythromycin and gentamicin resistance. Additionally, ability of GBS isolate to produce strong biofilm exhibiting a correlation with penicillin resistance. But we just know here, we did not look at the molecular mechanism, because if you remember earlier slides, I told you there could be mutation in the gene, there could be in the efflux pump or several others. Okay, we don't know till now what is the molecular mechanism of this penicillin resistance in group B isolate. So we are planning to do that. So right now we are working on the human peptide, because I told you antibiotic resistance is a great concern. So we were looking from some other alternative to treat this infection. So we have a collaboration with the Germany. So what they do, they have this human peptide library. So first thing you should know how they construct this library. So like the dialysis patients, okay, they do come for the dialysis in the hospital. What they do, they collect this dialysis waste and they make the whole library. So they do cation exchange chromatography and then reverse phase chromatography, because we are looking something which is positively charged, which can bind to the negatively charged bacterial surface. Okay, so we wanted to know here. So they have a huge number of the body fluids, like they have from saliva, from the milk, from the tears, from placenta, you name it, and they have a whole library from different organs. So we are screening here hemofiltrate library, which is collected from the dialysis patient, and we wanted to know whether these fractions, they have antibacterial or antibiofilm activity. Until now we have screened these whole like 450 fractions against different serotypes of group B streptococci, and some of the fractions we found they have a antibacterial and antibiofilm activity. So we can summarize it. Antibiotic susceptibility pattern, they are GBS is actually susceptible to penicillin, which is reported, but we found rare cases of penicillin resistance. biofilm forming ability. Many GBS strains, they can form biofilm, and biofilm forming GBS show high resistance to antibiotics, and penicillin resistance may be linked with enhanced biofilm formation. So, just coming to the some fun part, why did the bacteria join the biofilm? Why we live in a specific group? Because we don't want others to come into our group, isn't it? So same with the bacteria. They make their own biofilm. They don't want other bacteria to come into that. So bacteria and biofilm are like introverts at a party. They huddle together and don't let antibiotics crush their clique. So we can put it like this. This is a strong biofilm, like a huge castle. Weak biofilm, not so much. So it can protect bacteria from any antibiotic attack. Okay, thank you. So I'm done with my presentation. Any question? देखो दो बातें या तो बिल्कुल समझ में नहीं आया. हाँ, can you be loud please? हाँ.
My question is that the GBS, how are they actually getting antibiotic resistant?
How they are, so that's what I told you, we don't know. We have to look at the molecular mechanism. So we have a PB2 gene. We can go and sequence it to know whether there is a mutation in the gene or there is a mutation in the efflux pump. So for that we have to go into more molecular detail, but we don't know here.
Okay.
Yeah.
The mutation can be a problem. Mutation, it can be mutated genes.
It could be, yeah. Yeah, it is possible, yes.
Are there any prevention about that?
Yes, in other, so I don't know about our serotype, but previously it has been shown in other GBS clinical isolates that if they do have antibiotic resistance, there is a mutation in the PBP2, which is a penicillin binding protein gene.
Okay. Thank you, ma'am.
Any question? बच्चे सो गए सर. Okay, thank you so much.
Okay, thank you, Dr. Pooja, for the nice talk. And I think most of the students, they are already now have some knowledge on the biofilms, and especially she has taken the case of the Streptococcus species, different species of Streptococcus which are prevalent in India. So just to honor Dr. Pooja, I invite Professor Bhunia, Professor Arun to just felicitate our resource person. कोई नहीं, आओ आगे आओ.
हाय एवरीवन. मेरे थोड़े है ना लैंग्वेज बैरियर है, इसलिए मैं हिंदी में बात करूँगा. सॉरी फॉर दैट. माय नेम इज़ नवनीत कुमार एंड आई एम फ्रॉम हनुमानगढ़. आई हैव डन माय एमएससी इन बॉटनी और अभी मैं CSIR नेट के लिए तैयारी कर रहा हूँ. और अब तक मैंने मशरूम के ऊपर ट्रेनिंग की है, मशरूम प्रोडक्शन से रिलेटेड. बाकी 4 नेशनल कॉन्फ्रेंसेस अटेंड कर चुका हूँ मैं और 2 इंटरनेशनल कॉन्फ्रेंसेस. बाकी दैट्स ऑल. सर अभी मैं तैयारी कर रहा हूँ स्कॉलर के लिए.
I self Abhay Singh Jankaut. I am from Jaipur. I pursuing MSc Medical Laboratory Technology Microbiology Palwal Haryana. I pass my graduation in Dr. SN Medical College Jodhpur. My hobby is playing cricket.
Good afternoon, everyone. My name is Deepak Chaudhary, and I have done my bachelor's from Sri Vishwakarma Skill University, Dudhola Palwal, Haryana, and now I am pursuing MSc MLT Microbiology from Sri Vishwakarma Skill University, Dudhola Palwal, Haryana. सर हॉबी तो बहुत सारी हॉबी है. सर वॉलीबॉल भी खेल लेते हैं, क्रिकेट भी खेल लेते हैं. पहले तो प्लेइंग में तो कई सारी हैं और वॉचिंग मूवीज. यही सब है सर. हाँ.
Good afternoon, sir. Good afternoon, everyone. Myself, Sambhu, currently pursuing my MSc MLT Microbiology from Sri Vishwakarma Skill University. Deepak is my batchmate. And my hobbies are playing badminton and playing cricket. If I talk about my little achievement, then I have 3 book chapters which have been published, and I have attended 1 international conference where I scored first position in Galway University, and on the topic of prevalence of scrub typhus in Haryana. That's all.
I am Roshan Kumar Jha, PhD. I am Roshan Kumar Jha, PhD scholar of Department of Nutrition Biology from Central University of Haryana, and my research topic is multifaceted chemoinformatics study to characterize the bioactive compound of finger millet. Basically, my research work on bioinformatics, and I have already published 2 research papers in reputed journal. My hobby is playing cricket. मैं खेलता हूँ सर.
Good afternoon, everyone. Myself, Anita. I am a PhD scholar from Department of Nutrition Biology, Central University of Haryana. And my PhD topic is extraction and characterization of organic nanoparticle from Vigna aconitifolium and check its nutraceutical delivery. And I have done my masters in Department of Zoology, Kurukshetra University, Kurukshetra. Thank you. My hobbies is spending time with my son.
Very good afternoon, everyone. Myself Jyoti, and I am PhD scholar in Department of Nutrition Biology. So basically my topic is related to formulating dual millet fermented product targeting Alzheimer's and neurodegenerative diseases. So another thing, my hobbies includes yoga and helping other peoples, spending time with friends, traveling, photography. Thank you.
Good afternoon, everyone. I am Aheli Shamanto from West Bengal. I am an undergraduate student. Currently, I am in my final semester microbiology from Vivekananda Mahavidyalaya, the University of Bardhaman, West Bengal. And if I talk about my hobbies, I used to be an active participant in any cultural programs, but not continued anymore. And I'm an fitness enthusiast. Thank you.
I'm Boshudha Laha from Raniganj Girls College, KNU University. Recently, I'm pursuing BSc Microbiology, and my hobby is reading storybooks.
देख के इसीलिए बोल रहा हूँ, वो याद ही नहीं, सच बताऊँ मैं आपको, कई दिन हो गए देखे ना ना, अपने बारे में तो याद है, टॉपिक याद नहीं है ना। होता मेरी Myself Prasan Chaudhary, I am pursuing my PhD under Dr. Ashwani Kumar in Department of Nutrition Biology, and my topic is the determination of nutraceutical virtue of bioactive peptide of pearl millet through multi-pronged computational and structural approach. हाँ इसलिए याद नहीं है. And my hobby is to playing cricket. और भी बहुत सारी हॉबीज़ हैं अच्छी अच्छी, लाइक घूमना इधर उधर. हाँ जनरली like in every month I visit to different temples. ये थोड़ा सही है, बाकी तो पढ़ते रहते हैं, काम है.
Good afternoon, everyone. My name is Monica Chaudhary. I am pursuing PhD from Manipal University, Jaipur. My research topic is Parkinson's disease, broad topic. This is neurodegenerative disease. I haven't done anything right now, but I wanted to do something good in research. My hobby is playing badminton and spending time with senior citizens. Thank you.
Good afternoon. Myself Jashi. My BSc is completed from M.Sc. Dayanand University Rohtak, and PG is complete from IG University Mirpur, and pursuing I am a PhD scholar.
Good afternoon, everyone. Myself Sheetal, PhD scholar from in Zoology Department at CBLU Bhiwani. I have completed my masters from CBLU. My hobbies are reading books and listening music.
Good afternoon, everyone. My name is Dipesh Beniwal. I am from Sirsa, and now I'm pursuing my masters in biotechnology from Manipal University, Jaipur, and my hobby is playing football. Football, sir.
Hello everyone. Myself Deep Adhikari. I am come from West Bengal, and currently I am pursuing my BSc Microbiology from Kazi Nazrul University. And my hobbies are playing games and movies, watching movies, animes. That's a simple statement, but I am less interested in theory parts and more interested in practical parts. Last line. सर मैं थोड़ा सा theory portions में less interested हूँ, और less interested in theory parts, and that's a simple statement.
Good afternoon, sir. Good afternoon, teacher all. My name is Rupak Patra. I am from West Bengal, and I'm currently pursuing BSc second year from Kazi Nazrul University. My hobbies are watching movies and reading some books, especially story books. Now I am preparing for competitive exams for getting a good college in MSc, for pursuing MSc. Thank you.
Good morning, everyone. I am Shubhadeep Dotto. I am from West Bengal. Oh, I am from West Bengal. I am pursuing my microbiology honors degree from Banwarilal Bhalotia College, which is affiliated to Kazi Nazrul University. And I have participated in a few conferences and a few workshops, but they aren't directly connected to the microbiology. This is the first time that I got this precious chance to participate in this workshop, which is directly related to core microbiology topic. So thank you for providing me this precious chance. I'm really grateful to the authorities and PhD scholars for supporting us. My hobby is playing a little chess and sometimes watching movies. That's it.
Hello everyone, I'm myself SK Hassan Raza. I am from West Bengal and currently I am pursuing my BSc in Biotechnology from the Bardwan Institute of Management and Computer Science under the affiliation of Bardwan University. And my hobby is experience new activities like food, places, and but actually मतलब कुछ एक्सपीरियंस करना, एक्सप्लोर करना जो मैंने पहले नहीं किया था, तो दैट्स माय हॉबी. क्या सर? हाँ सर, आते वक्त बहुत मतलब हाँ कल हम निकले थे कैंपस घूमने. हाँ कल हम शाम को निकले थे कैंपस घूमने के लिए. हाँ यहाँ पर नीलगाय देखा, पिकॉक्स देखे. कैंपस भी सर बहुत बड़ा है, हमको घूमते घूमते पैर में दर्द भी हो गया थोड़ा सा.
Good afternoon everyone. I am Kushal Mollik. I am from Vivekananda Mahavidyalaya. Now I am pursuing final year in BSc Microbiology, and my hobbies सर गाना सुनना, गाना करना, ड्राइंग करना अच्छा लगता है और घूमना अच्छा लगता है और भी बहुत सारे हैं.
Good afternoon, everyone. I'm Moinak Mondal. I'm from West Bengal, and I'm currently pursuing BSc Biotechnology under the government of University of Bardhaman. And these are actually all we all came from the same place, and these are actually my batchmates. Yes, sir. And yes, sir. And my hobby is actually playing video games and all. Thank you.
Good afternoon all. My name is Sudan Tiwari from Devariya. I am pursuing PhD in microbiology from Bundelkhand University. My hobby is playing chess and traveling spiritual places and other places. Thank you.
My name is Nikhil Viskarma, and my bachelor's complete from Kanpur University. After my bachelor's, my master's completed for same university, which is Kanpur University, and currently I am pursuing a PhD from Bundelkhand University, and my hobbies are using my free time in traveling, playing cricket. Thank you.
Good afternoon everyone. Myself Vansh and I am from Haryana Palwal, and I am currently pursuing my bachelor's in vocational medical lab technology and second semester, and my hobbies are playing cricket, volleyball, and I love to play video games. Thank you.
Hi everyone, I am Lokesh Kumar and I am doing my bachelor's degree from Sri Vishwakarma Skill University in MLT, and currently I am in the first year, and my hobby is reading spiritual books.
Good afternoon, everyone. Myself Shivamuja. I am from Delhi. I done my bachelor's of science in medical lab technology from Ansal University, Gurgaon. Then I worked as a blood bank technician for 2 years. Then I enrolled in masters of science MLT in microbiology. I am in first year. My hobbies are traveling or reading historical books.
Good afternoon, everyone. My name is Divya, and I am a master's student, and I am pursuing my master's degree from Department of Biochemistry from this university itself, under the guidance of Dr. Maruti Mulaka. And currently, I am doing my dissertation work. My dissertation work is on pulmonary fibrosis, and my hobbies is listening music.
Good afternoon, everyone. My name is Yogita. I have completed my master's from this university in environmental science, and my hobbies are reading novels and singing. So, reading novels and singing. Yes, sir.
Good afternoon. I am Sani from Delhi. I am MSc MLT from Sri Vishwakarma University, Dudhola, Palwal, and my poetry. Thank you.
First of all, thank you so much for giving me this opportunity to introduce myself. My name is Urmila from Palwal. I am doing BVoc in MLT from Sri Vishwakarma Skill University in first year. My hobbies is listening music and making drawings.
Good afternoon, everyone. My name is Jyoti from Charkhi Dadri, Haryana. I'm pursuing my master's in MLT Microbiology from Sri Vishwakarma Skill University, Haryana. My hobby is watching cartoons. Thank you.
Good afternoon, everyone. I'm Savita Bharti. I am student first year in BVoc MLT from Karma Skill University, Dudhola. Hobbies badminton and talking about something spiritual and also knowing about that, and also include in my hobbies are fighting with my siblings, what is and what is I really miss. Yeah.
Good afternoon, everyone. My name is Nikhila Khola. I am pursuing PhD from Department of Biochemistry CUH, and my research topic is to study the effect of dietary flavonoids in neurodegenerative diseases. I love cooking and maintaining a kitchen garden.
Good afternoon, everyone. My name is Prashant, and I am currently pursuing masters in biotechnology from Chandigarh University. And talking about my work, I currently am writing a chapter on conversion of cereal waste into biofertilizers, and I have done a workshop related to cordyceps as well. So yeah, thank you so much.
Good afternoon, everyone. My name is.
So my hobbies is writing and playing football. Yes, sir. Yeah, writing and playing football.
Good afternoon, everyone. My name is Vaidehi, and I am currently pursuing MSc from Biotechnology Department from Chandigarh University. Right now, I am working on a project of sodium hydrolysate, and my hobbies are singing, and I like to host events also. I have done some events, and I like to write also, and I like to dance. Thank you.
Good afternoon, everyone. Myself Pratiksha Latoria. I am from Bundelkhand University, PhD scholar. I have completed my master's and bachelor's from Integral University, Lucknow. And my hobby is traveling and cooking.
Good afternoon to everyone. Myself Dipanshu Punyani from Panipat, Haryana, and currently I'm a first year PhD scholar under Professor Gunjan Goyal, and my hobbies are working out in gym and playing cricket.
Good afternoon, everyone. My name is Khushbu Negi, and I'm currently working as a GRF under Dr. Pooja Yadav. And I had done my master's from Central University of Rajasthan, and my hobbies are playing basketball, singing, dancing. Thank you.
Hello, good afternoon everyone. Myself Neha Kavar Nirvan. I am a PhD scholar in the Department of Microbiology in Central University of Haryana. Broadly, my topic would go towards exploring the potential of plant endophytes. My hobbies include listening to music, watching movies, and reading good stories. Thank you.
Good afternoon, everyone. My name is Surbhi. I've done my master's in microbiology from Central University of Haryana. Currently, I am working in a project under Dr. Gunjan Goyal on the development of GABA-enriched probiotic millets with the nutraceutical potentials. My hobbies include gym, fitness, yoga, and I love reading novels in all genres like thriller, suspense, rom-coms, mythological.
Good afternoon, everyone. Myself Jyoti Yadav. I am pursuing my PhD from Central University of Haryana. My research topic is isolation, characterization, and application of asparaginase from efficient microorganisms. My hobby is playing chess and spend quality time with my family.
Good afternoon, everyone. I am Jyoti Singh. I am first year PhD student in Central University of Haryana, Department of Microbiology, and my hobby is drawing and painting. Thank you.
Good afternoon, everyone. My name is Ayush Mathur. I am a PhD scholar in Central University of Haryana, and I am from Jaipur. My hobbies are photography and playing chess. Thank you.
Good afternoon, everyone. Good afternoon, everyone. My name is Nayan Rishi. I am a PhD scholar in Central University of Haryana under Professor Gunjan Goyal sir. And my topic of the PhD is bioprospecting probiotic yeast from traditional fermented beverages. And my hobbies, I am a cynophile, a person who loves the dog.
Good afternoon, everyone. I am Rohan Poddar. I am currently pursuing my master's degree from this university in Department of Microbiology. I am currently working as a dissertation trainee under Professor Gunjan Goyal sir. My topic of dissertation is antimicrobial resistance in commercial probiotics. As for my hobbies, I dabble in sports. I have played 2 national zones for this university in basketball, football. I also like to cook. Thank you.
Hello, good afternoon, everyone. I am Deepak Kumar. I am from Bihar. I have done my graduation from BHU Varanasi. Currently, I am pursuing doing dissertation under the supervision of Dr. Pooja Yadav ma'am. My topic is antibacterial activity of Enjetamansi of gram-positive and gram-negative bacteria. Thanks. My hobby is playing cricket.
गुड आफ्टरनून एवरीवन. मेरा नाम रोहित फोगाट है. मेरी ग्रेजुएशन दिल्ली विश्वविद्यालय से हुई है और मेरी पीजी एमडीयू रोहतक से है और मैं 2021 से प्रोफेसर गुंजन गोयल की लैब में पीएचडी स्कॉलर हूँ. होपफुली दिस विल बी माय फर्स्ट फाइनल ईयर.
Hello everyone, my name is Yatin Kumar. I am from Delhi. I have done my graduation from Delhi University Venkateshwar College. I have done my masters from HNB Garhwal Central University in microbiology. Currently I am a 4th year PhD scholar and my PhD topic is identification of heme transport system in Vibrio alginolyticus. Mostly my field is molecular microbiology and I have my hobbies are working out in gym and dancing and rapping. That's it. Thank you.
Hello everyone, I'm Shopan. I'm from Kolkata and I'm pursuing my PhD under Professor Gunjan Goyal and I'm working on GABA producing probiotics and I'm starting my 3rd year and my hobbies include watercolors, and I'm good in debating, and also listening to English classical music and cooking. Thank you. And we would also like to know sir's hobbies.
That's a good point. So when you introduce each other, I think in this type of community, it's always good to talk about you because you need to know each other. And we are all in academia. Education is our primary goal, right? You'll be spending a lot of time in the laboratory, doing research, reading, research articles. And so obviously you'll be spending a lot of time. But don't consider your life to be focused only on science, because that's going to be your life, right? Whether what position, whatever job you do, that will be your primary work. But always you need to have a way to get away from that, right? If you are always focusing on doing these things, and you are not going to have a fresh look at it. So that's why always you need to get away, step away from it for a while. How do you do that? Follow your hobby, right? I mean, asking you what's your hobby, you are reinforcing that I like chess, I like music, I like cooking, I like reading book, I like, online stuff. Nobody said that, I spend a lot of my time looking at the phone. Anybody said that? That's not your hobby? See, I walked around the campus. Every time I see any of the students, I'm not necessarily you, maybe I haven't seen you, they are all on their phone all the time. I mean, I haven't seen them, they are doing something. Even two people sitting next to each other, they're both on their phone. They are maybe texting each other. That's their hobby. They don't want to communicate, all signaling, right, in the biofilm. So they are not doing that, quorum sensing. So hobbies are very important. Always pay attention to that. Try to make time to do your hobby or focus on your hobbies. It's extremely important for your personal growth. Always pay attention to that. And if you are happy, everybody surrounding, nearby, they are also going to be happy. So the hobbies are very important. So my hobbies, I know you put me in a spot. When I used to do a lot of painting and drawing. And so the book you received, or the textbook, if you check it, a lot of artwork. I used to do, when I had a lot of free time, I used to do that. Painting and drawing, and that was my growing up hobby. When I got into science, and I'm trying to find an outlet, how do I use my hobby into this science? So when I started writing these books, I usually use my own drawing, sometimes hand drawing, sometimes digital drawing, but nonetheless, these are all scientific drawing. So you might find a lot of those artwork in my book. So that's still my hobby, and unfortunately, I don't have a lot of time to do that hobby. My daughter somehow picked up that, probably it's in the gene. So she's a very good painter. I mean, she's much better than me. And she does a lot of those artwork exactly like mine. I was kind of surprised. And one thing I haven't been able to fulfill, hopefully I'll do that someday. Each time she come home from college and ask me, Dad, can I draw with you? I haven't done that. But say, let's do painting something together. I haven't been able to do that. But hopefully someday I will do that. But that's my hobby. And I know some of you, painting and drawing, that's good. That's a good hobby. It relaxes you a lot. But every hobbies are important. Try to follow through besides science. So that's what I do.
Okay. Okay, thank you, Professor. Like Sir said that, okay, the timing of the office should be restricted. And all of you know, all of the students, they know that I leave this office by 5. Because work is never ending. आप घर पे भी जाओगे तो भी आपके पास काम रहेगा. आप 6 o'clock तक बैठोगे, 7 बजे तक बैठोगे, 8 बजे तक बैठोगे. Work will never end. So I used to leave this place at 5, because to get mental distress, or you can say that how to remove the stress, I spent time with my family. Regarding my hobby, I used to draw pictures. So abhi to time nahi hai. So I just draw pictures of the son when they have some homework from the school that they have to draw something like this. So I used to prepare the charts, their drawings and everything. Thank you.
Now you can go and enjoy.
अभी तो कोई हॉबी नहीं है सर, बट या डांसिंग I won state level awards in dancing actually, and debate and then poetry.
Good afternoon, everyone. I am Himanshu Singh from Agra, and currently I am pursuing my PhD under the guidance of Dr. Pooja Yadav from here at Central University of Haryana. And my hobby is listening music. बस बहुत है इतनी है. Watching movie and Taj Mahal also.
Right? I think that's one of the questions always you are asking, right? So let's explain, or let's try to find out the mechanism behind persistence. Also, towards the end of my lecture today, I'm going to talk part of our research data to show you how we study the diseases. Okay, so persistence of pathogens. Factors responsible persistence. So again, we are telling the same story in different ways. We mentioned a little bit about viable but non-culturable state. We'll examine a little bit that state of so these type of cells or cell population, they also maintain a called viable but non-culturable state, and that also are a community of persisters. Okay, I tried to explain that last time. What is viable but non-culturable? Then biofilm formation, obviously, those cells try to form biofilm, or within the biofilm community, they are also persisters, right? Quorum sensing, of course, that plays an important role, and spore formation. That's also a strategy for microbes to serve, maintain as a persister. Of course, we are not going to talk much about the spore formation, but that's a natural process for some of those microbes like Bacillus or Clostridium. Many of these properties actually start with stress, right? Biofilm is a stressful condition, or bacteria going to a persister phase, that's because of the stress. Okay, so that's why we're going to spend a little bit of time. What are the stress means to you? I mean, you can apply that information to yourself. So if you are under stress, are you more productive or less productive? A lot of times, negative effect, right? You become less productive. So that means you go through some kind of dormancy, or kind of like a less active phase. So some of those stress can make the organisms going through that transition to become more persistent. So we're going to examine that. So here we're going to talk about stress response and adaptation, and then effect of pH, salt, acid, temperature, any of those standard, physical parameters that can impact the cellular physiology that leads to persistence. And I mentioned a little bit about viruses last time. So viable but non-culturable state. Let's look at those populations. Organisms that are no longer culturable with standard non-selective culture media. So this terminology VBNC is critical for microorganisms. As I mentioned yesterday, that you take a sample, you try to grow them in the culture media, and then you find out there is no growth, right? If you change the condition, then after some time, then you start growing. Means that same sample behave differently. So that's called the viable but non-culturable. So what happens? Their metabolic activity, the metabolic process slows down, or they may be producing certain enzymes that's needed for their to maintain their viable but non-culturable state. Maybe they are not synthesizing enzyme, or they need enzyme that's required for harvesting energy so that they could be metabolically active. But in this VBNC state, some of those functions may be arrested or stopped or halted or not at all. Everybody got it? So some of those enzymatic activity slows down or stops. Nutrient uptake, they may not be taking uptake nutrient, therefore that also slows the metabolic process. And other aspect, also a lot of times the cells change their morphology. Okay, that's kind of fascinating. The cells, they could be rod-shaped cells, and then suddenly they become round cells. Okay, but we are so much trained to study microorganisms under microscope, right? We know some organisms are always rod cells, right? Or some cells are always round cells. or small cocci, okay? So that's our knowledge, that's our familiarity. But as soon as you see certain cells change their morphology, then you get already, I said, oh, it doesn't look like it's a salmonella, or it doesn't look like it's E. coli or listeria, because the shape changes. That I'll talk about that. And so those are some of those attributes that relate to the viable but non-culturable state. So some of those examples of microorganisms which goes through BBNC state: Campylobacter, E. coli, Listeria, Salmonella, Shigella, Vibrio. That's original study actually started with the Vibrio. So let's look at a little bit about the stress response. This looks a little bit complex. Let me simplify it. So there is this called level of stress. There is a degree of stress, right? Some stress could be high level of stress, right? We all know that. Or some could be moderate, or some could be low level stress. So depending on the degree of stress, you may see different effect on the bacteria. So let's say this is a normal growth range. This is the, I want you to pay attention to the left side of the graph. So this is the level of stress. Some stress could be sublethal, means it's not killing it, but it keep it sublethal. They can regenerate or come back. And some of the lethal means they are going to die, right? We don't worry about that. You introduce some stress. So for example, heat, right? You started heating bacteria or exposing them to 50 degrees, for example, 50 degrees C. So you will see that some cells, it's going to be lethal to them, but some bacteria, it may not be lethal. So that will be sublethal. Everybody clear? So under 50 degree, which is not normal for that bacteria, so they are going to go through stress. And so what happens in this sublethal, if it is that 50 degree, so some cells will have an injury, okay? The cells will be injured, but they are not dead, but injured, okay? And then there is some stress adaptation. If it is a low level stress, not sublethal, there will be some level of adaptation. And we always know that we can adapt, right? Even the temperature going up here, we can adapt. We are not dying, right? But we are highly stressed. We cannot function properly, right? We cannot think properly if we are under stress. So that's where we can adapt. But when sublethal, that injury, this can be repaired. It's not that we are irreparable. We can repair ourselves. So this is a sublethal injury. But if you look at this window between lethal and sublethal, and this is where the bacteria can get into non-culturable state. So it's not dead, but they are like a unresponsive. It's like a totally dormant. Are you getting it? It's like a zombie. You've seen these zombie movies, right? So the dead coming back to life. So they are staying dead all the time, and suddenly they become alive. And so kind of like they fall in the grand scheme of stress response. And of course, like I said, the dead cells, you cannot culture them. They are always dead. They are not going to be recovered. So just give you that idea. Now let's look at it. What is this persister terminology and persister phenomenon came? So the BBNC states, like in this plot, if you look at the bacterial cells, if there is environmental stress, and these organisms are going to be part of this stressful community, then when they are under stress, some population can start forming biofilms. Now start thinking the reverse way. If there is a stress there, and that may promote bacterial biofilm formation, and then some population within that, okay, they would be called persister cells. Means they are not responding, they are kind of dormant. They could be BBNC cells, okay, and they are just become persister. So why that happens? So nutrient limitation is one factor. Changes in sodium chloride, levels of sodium chloride in the water, especially in the ocean water. That's where we see a lot of BBNC cells. Exposure to hypochlorite, shifts in temperature. So a lot of these factors contribute to the BBNC state. Think about it now, that when microorganisms, we are trying to kill them by a number of different ways. And some of these happen to be some of those factors. So obviously, we can create some of those organisms to undergo VBNC state. That makes sense? Because we are trying to kill them, right? We are going to induce injury, then followed by lethal treatment. But when we are not perfect in providing that environment, or even in the nature, that can happen. And so the organisms can go through VBNC state. So this one is just saying that VBNC cells, we can resuscitate them. Then you have to reverse it, right? They are not dead. They are dormant. They are staying in a place. They are not responding to natural conditions. But if you shift the temperature, okay, so normal temperature is 37, and if you shift it to say lower direction, 30 degree, maybe that's favorable for that organism. Now it can wake up, can come back to life. Maybe that can grow. Now they can reverse back. Also, return to nutrients. So if there is a nutrition starvation, now if you give them food, they can come back to life. So how about the catalase? Sometimes we produce hydrogen peroxide. Like for example, in the macrophage, when the bacteria are engulfed, macrophage produces hydrogen peroxide. And hydrogen peroxide essentially going to kill the organism, right? But if you make catalase, you can neutralize the hydrogen peroxide, okay? So that means if we add catalase, now you can neutralize hydrogen peroxide action, now the organism can come back, okay? And sodium pyruvate also, it scavenges some reactive oxygen species, so the organism can come back to life. So these are some of those factors. You have to add it to the media, and then if you have a VBNC cells, they can come back to life. And that's essentially what we do. So in order to avoid being VBNC state, you can resuscitate. Everybody understand the resuscitation? Resuscitation might, like if you suddenly lost your consciousness, right? And then or you were dying, right? Or you had a heart attack, and so your heart stopped. And so people start to resuscitate them, right? They give mouth-to-mouth breathing, right? They do the whatever called the heart pressure, right? To bring the heart back, and that's called resuscitation in real life. And so the organisms which undergo stress, undergo injury, you need to bring them back from VBNC state or injured state to healthy state. And that's what we call resuscitation. And so resuscitation state needs to be followed through. Okay, so in this one, if you have active cells, we know they are going to multiply, right? If you have a persister cells, those who are dormant, those who are not responding to any nutrient, you need to bring them to life, right? So that's why there is a step in between dotted line. We need to return to the active condition. If you bring them to active state, now they are going to multiply. You can culture them, right? So this is, then if you have a VBNC state, VBNC also part of the persister community, right? And so now you can make them to persister cell, and then become active, and then they start to grow or multiply. So all of these factors you have to understand how to bring them back to normal conditions, so then you can grow them. Okay. So persister cell, you can easily study it. I think there are a lot of these nice review articles came out. So if you take a population and expose to antibiotic, for example, right? And you know that as soon as you expose to antibiotics, those who are resistant, they are always going to grow in that condition, right? So let's take that population right here. Then those who are susceptible, they are going to die right away, right? But there is a population which is not going to die, but this is a tolerant. Tolerant also a little bit different. They are not resistant, but they are tolerant. They can tolerate up to a certain level. Okay, but eventually, as you increase the concentration, they are going to die. Tolerant is all dependent on the concentration of the antimicrobial agents or the antibiotic. But there is this population after antibiotic treatment, they are not tolerant cells, they are not susceptible, they are not resistant, but they are persistent. And that happens when you treat the patient with antibiotics, and these persistent cells can eventually come back. Very clear? And this is what we've seen. They are not resistant population. So something happens to this population of microorganisms or bacteria, they tend to convert into called persistent cells. And so to understand the mechanism of persistence, you have to really understand the molecular control, the regulation. Okay, I'll try to explain you a little bit, but probably it needs a lot more reading, but try to simplify it. So persistent cells, this has been a long history. Okay, for I think 1942, that's when the Hobby actually he discovered the phenomenon. This individual, Joseph Bieger, actually named these small numbers of bacteria that survive penicillin treatment, and those are called persisters. The actual terminology coined from 1944. And then over the years, there's a lot of work being done, and they are trying to understand the mechanism. And this actually came from this article, review article, published actually 2024, and talked about it. So I try to simplify it. This is really a complex structure, and why certain cells undergo persistent stage, and how do they do that? Okay, why this happens? So there is a two phenomena. We call it toxin antitoxin phenomenon. Okay, in cellular system or microbial cells, the toxin antitoxin works cooperatively. Okay, when you say toxin, means it's going to degrade something. Toxin has a negative effect, right? That's the simple way I try to present it. So if you take a toxin, it has a protease activity, it's going to degrade messenger RNA, for example, right? And so there won't be any protein synthesis. So if you don't have that protein synthesis, that protein is essential for your survival. So let's say protein important for substrate utilization or enzyme. Okay, if that's not produced, then what do? You cannot use that enzyme to harvest energy. So therefore, in that case, your system not going to work. Means it's going to slow down. Then antitoxin is kind of like a balance. It has some regulatory element, and so this antitoxin-toxin effect always kind of working together. Okay, so toxin is going to try to degrade it to make it cells like a persister. I don't have enzyme, so I cannot use the substrate. I'm not dead, but I can maintain my life. But antitoxin, when it comes, it will contradict that. And as soon as that antitoxin starts working, then the persister cell can become more healthy cell. Okay, that's the simple way I can present it. But it's all enzymatic action, how they manipulate their messenger RNA for production of say enzyme or toxin, which are essential for their survival. Got it? So I want you to read it. It's a little bit complex phenomenon. This is a nice review article, talked about how this works. Okay, other central molecule play important role. So persistent cell formation by toxin antitoxin system. So if you say that genetic basis of persistent formation, this toxin antitoxin I just mentioned, that helps maintain the dormancy state and controls the cellular process. Okay, you are going to get this slide, so obviously you'll be able to read it up more. And then there are different types of inhibition happens: type 1, type 3, then type 2, and then type 4. These are all called antitoxin toxin effect, and that's how it mechanistically helps the cells to maintain persistent state. Okay, this is a simple way I try to present it. Okay, so strategies to control persistence. How do you control these persistent cells? Obviously, they are a problem, right? You try to kill them with antibiotics. Some of them are not killed, but then they are still maintained, right? So how do you manage them, control them? And so this review article, again, it really took nice approach to say, okay, you need to have a combination therapy, right? So for example, you need to have a anti-persistent drugs together with antibiotics. So you can just say, okay, I can give you a penicillin, but penicillin may not kill that cell, right, alone, which is going to maintain a persistent stage. But then can I have anti-persistent drugs together with the antibiotics? Then you will be able to remove all those cells. Here they used antibiotics and then some drugs, again same mechanism, and then drugs are sensitive to persister. Immunomodulators. So there are a number of, you can use, say membrane damage component which can help the drug to penetrate through the cells which are resisting certain antibiotics. Okay, so all of these combinations can be used to help make the cells persistent. So again, I try to give you some idea, but you need to read up a little bit more because it's not possible to explain everything within this short period of time. Just trying to give you an idea so that when you go and get a chance, read up some of this. Okay, so as I said, stress become one of the major factor in inducing the cellular process. Okay, whether it's a VBNC state or persistent state. So these are the common factors, right? We talked about temperature, acid, antimicrobials, then of course the biofilm. When the cells form biofilm, part of the community become persistent. Okay, so cell morphology, like I mentioned about that, the cells change their morphology. Look at the Vibrio, how they look like. They are curvy rod, short rod. Okay, that's how the Vibrio look like. This is a Vibrio cholera on the left. But when they get into VBNC state, this is how they look like, round cells. Have you ever thought that rod-shaped cells become round or coccoid? And so when you look under, we did some work on this a way back, and we looked at the VBNC Vibrio, they are round cells under microscope. So when you look at it, then you said, no, it might be something else. You never think that this is Vibrio. But if you reverse the condition, so ideal environment, and that's what become a rod-shaped cell. So when you are testing the water, say for example seawater, that's where you see a lot of Vibrio cells, and you bring the water into the lab and then do the microscopy, you might find this type of cells. This is the majority population. Then this become rod-shaped cells, and you can reverse that. You can induce VBNC state, and then rod-shaped cells become round circular cells. Okay, so those sort of things you can manipulate. Okay, what about this? So you can see these small cells, the shorter rods, right? What is this cell? If you are looking under microscope, what would be your first impression? It's a contamination, right? If, as a microbiologist, always you used to see certain uniform population. If you see a different shape, size in same population, you immediately think, oh, this must be something else. So actually this is salmonella. Okay, these are salmonella cells, and some salmonella decided not to divide. So this become a long rod cell. If I have seen a bacillus under microscope, bacillus make a long chain. Okay, now salmonella decided to become a long salmonella cell. Very unreal, right? And you will think that immediately it's actually a contamination. Okay, now answer my question. Yesterday I discussed about cell wall, right? I talked about cell division a little bit. So what do you think is happening in this cell? Why this cell became a long rod-shaped cell? When we see single cell, what does it mean? Single cell become two cells, right? And they separate from each other. So then we see these two single cells, right? And this is how they divide. That's their natural process. So during cell division, what happens? I'm looking at, so yesterday remember peptidoglycan structure I was showing you, and I showed you N-acetylmuramic acid, N-acetylglucosamine, how they are linked and how they have cleaved. There are enzymes, right? Well, there is a peptide there, peptide bridge, but between N-acetylmuramic acid, NAM, NAG, there one beta link, right? And did I mention some of those enzymes which cleaves those? I was using Staph aureus as an example. So what is happening with this stress? So those enzymes are not working. They make a lot of those autolytic enzymes. That's what helps them to divide. They become a single cell. So if your stress inhibit those enzyme synthesis, cells are not going to divide, right? So apparently this long cell, somehow some of those enzymes are not working. Stress can do that, right? Stress can, that's the whole idea. The stress factor can control the expression of some of the gene products which are responsible for, in this case, cell division. Got it? Everybody clear on that? And so look at this. Filamentation caused by starvation. What did I say? Nutrition, right? Lack of nutrient. That can be long cells. Low temperature, low water activity, desiccation, preservatives. A lot of those factors can induce stress, and so the bacteria would behave differently. Everybody clear so far? Can you link the dots I've been trying to address? Okay, let's look at another organism. This is Listeria, which is very short rod cells, 1 to 1.5 micron long. And if you hit them, can you see this? These cells are long. And what else they are doing? Aggregating. Look at that when they grow them at salt, 5.5%. If anyone looking at these cells, you will never think these are Listeria, right? Actually, we are used to seeing Listeria like this short rod. But if you grow them under 5.5% sodium chloride, which is pretty high, very high, okay, and this changes morphology and the shape. And if you look under that electron microscopy, look at this short rod, and now you can see these cells are joined, and look at this salt. I never seen Listeria become such a long chain. What happened again? Salt interrupting this enzyme activity, which is supposed to break it, separate the cells. That's not happening. So these are very common phenomena. This is just recently, we are working with E. coli, it's enterotoxigenic E. coli, and send this culture to my colleague, and we are trying to see their responsiveness to antibiotics. Okay? And so he said, well, let me grow it in the ampicillin because they could be ampicillin resistant. Have you ever thought this is E. coli? And then as soon as you remove the ampicillin, look at this. It reverts back. So you need to be preparing your mind that any time you change that condition to stress can change the morphology. Again, it's arresting a lot of those enzymatic action, and then cells become totally morphologically different. So that gives you a clue that why they are not going through BBNC or persister phase. Okay? But morphology is pretty dramatic. I mean, you can clearly see there is no question about it. Okay? All right, so that changes. Now, I think what I was trying to tell you about the role of, let's see. Okay. So next I'm going to look at example, tunicamycin. Let's use one antibiotic. Let's play with it. Okay? So antibiotic we add at concentration to inhibit microorganisms, right? Everybody clear? Okay, so what if we don't provide the actual lethal concentration? Lethal concentration means concentration that's going to kill all the organisms, right? But what if I provide sublethal dose? What happens to the microbes? Right? Because that happens, right? Sometimes we don't take proper dosing. And so does it affect the microbial behavior? So I showed you this yesterday, right? And how the peptidoglycan synthesis works, and then how the teichoic acid or wall teichoic acid synthesis. These are the two major gene products, right? These two genes help in the peptidoglycan synthesis, and taro is actually for wall teichoic acid synthesis. So tunicamycin, which is a small the antibiotic, it inhibits actually this gene. So therefore, this teichoic acid synthesis is inhibited. Okay, and I mentioned that wall teichoic acid also play important role in biofilm formation, right? So now, if I don't kill the organism, because if I kill the organism, I cannot study its effect, right? So I need to give a sublethal dose. So that's what I was trying to explain to you. Okay, let's say I gave a 2 low doses of tunicamycin to the bacteria and see how that affects the microorganism. First of all, we use Staph aureus and Listeria. So this is zero amount of tunicamycin is control. If you look at the cell morphology, it's still maintaining round shape, and you can see the cell wall and cytoplasmic membrane pretty clear. Okay. As soon as you add a low concentration of tunicamycin, 5 microgram, we consider as a sublethal, you can clearly see the cell wall structure started to disrupt, right? You can see the peptidoglycan structure is very thin, and that says totally different, right? So it's working in a partial level. So structurally it's begin to affect. And then if you look at the same happen with the Listeria monocytogenes, 0 versus 5, and you can clearly see the cell wall structure becoming very unstable, very disrupted, right? And so it affects what we propose. Now I want to see how that affect the morphology. Tunicamycin at low concentration. So this is cell morphology for Listeria. You can see it's normal, and as soon as you use 0.3 microgram level, and you can see this morphology begin to change. A little bit higher, you can see also again change, right? So you are seeing that bacteria are undergoing stress, and therefore their morphology is affected. Already I mentioned that it's going to affect the morphology. How about the colony? If you look at the normal colony, and as soon as you have antibiotics, the colony morphology also changes. There is one type of technology we developed called light scattering, and this is a healthy colony, and it creates this beautiful scatter signature. But as soon as you have a colony formation like this, the signature different. So it's telling you that cellular morphology, as it being affected by antibiotic and its structure, and then its phenotypes also changes. Same thing happened with Listeria monocytogenes. Same thing happened with Staph aureus. Okay, this phenotype change, and you can see the cellular presence with the different concentration of antibiotic. What about the biofilm, right? We mentioned that teichoic acid or wild teichoic acid is responsible for biofilm formation, right? So does it affect biofilm formation? Let's look at it. You can ask a number of ways. So in this case, we are just counting bacterial cells, and this is the crystal violet staining. I think we're going to talk about crystal violet tomorrow for the students. And so if you have a antibiotic to concentration, you can see that biofilm cells are reduced level. Biofilm here also reduced. And I mentioned this morning about eDNA. You can measure eDNA. eDNA synthesis also significantly decreased by antibiotics. So that means biofilm formation is affected. Just look at the images. Here, if you have a normal biofilm produced by Staph aureus, as soon as you have antibiotic, you can see it's dispersed and much lesser biofilm. So it clearly says tunicamycin affects wild teichoic acid, also peptidoglycan structure, and that also in turn affects the biofilm formation. So that means if at sublethal dose, if you are unable to kill them, you are going through these stressful conditions where microorganisms behave differently, they respond differently. Everybody clear on this? Okay. Then if you look at their ability to adhere to cause infection, I mentioned this morning again that adhesion is one of the characteristics, right? The microorganisms produce protein or carbohydrate to help them attach to surface, so that part of the infection model, and then they can cause infection. So if you have antibiotic at different concentration, their adhesion to cells, host cells, epithelial cells are significantly decreased, right? Same way with the Staph aureus, and while they are less toxic to those cells. And without going too detail, also we saw same phenomenon for Listeria monocytogenes. So the bottom line of this is that if the bacteria are under stress because of sublethal antibiotics, their infectious properties also changes, and also their biofilm characteristics. Okay, number of ways you can prove this, whether this affecting their virulence or pathogenesis. This is another way you can see if they regulate certain inflammatory pathway, and obviously you will see a decrease. And this is cytokine production. Like I said this morning, inflammatory cytokines TNF alpha, IL-6, their level also decreased significantly because they are not infecting at that level. But they are still pathogenic, but their degree of pathogenesis is decreased significantly. This is also for Staph aureus, this for monocytogenes. So just trying to point out that the structural components which are integral to the bacterial properties that help them to form biofilm and also contribute to pathogenesis. And if I used a high concentration of tunicamycin, would have killed the cells, right? Then I will not be able to study all of those things. So you can design your experiment to induce stress and find out stressful conditions still make an impact on the microorganism's ability to cause disease or infection, at the same time biofilm formation. Everybody clear on this? Okay. Let's look at other factors. That's some studies. And if the organisms undergo stress, I mentioned that sometimes they don't produce certain proteins. So this is a clear example. Very simple example though. If you take Listeria monocytogenes and all of these media, I'm not going to name all of them, pressure broth, UVM, all of this, all this media has certain antibiotics. Okay? Different levels, different antimicrobial agents. Everybody clear? Now I grow them. Let's look at their protein profile. If you grow them, same bacteria in one media, I found only one and these two major proteins showed up. Same bacteria grown in another one, I saw this protein and this protein. Another media, you can see this profile. And this is another media. So this is fascinating, is that bacteria, because of different antimicrobial component, they stop producing certain proteins. Maybe some of those proteins are important for adhesion or attachment or biological function. But just to point out, when the microorganisms are under stress, they stop making certain proteins. Everybody clear on this? Any questions so far? Okay. This is another example. In certain media, they will make a lot of proteins. You take Listeria monocytogenes. In certain media, they will make a lot of proteins. In another media, oops, just forgot to produce protein. If you are trying to detect this organism with this antibody, you are going to find negative. Or for running function. Some of them just never found any of these proteins. So the bottom line is that the stress can do a lot of different things to the cells. And no wonder they go through this type of transition. Okay, the diseases. Let's talk about the diseases caused by bacterial biofilms. I already showed you this slide before, right? You recall? And atherosclerosis, infection, endocarditis, all kinds of wound infection, cystic fibrosis, urinary tract infection, all kinds of diseases, right? We know that's responsible by biofilms, right? Now, this is also, you have seen this, dental plaque, mastitis, intestinal colonization, and so on. So, medical devices also you'll find this biofilm formation, right? And all of those we discussed. So, the wound infection, mastitis, we talked about it, right? The different types of organisms. So, these are responsible for causing diseases. So, biofilm also in the gut and the disease production, which is very important. Before I talk about the gut, the role of biofilm in diseases in the gut, first of all, one thing I want you to know, the normal condition. The normal condition is what? What you are, right? So if you look at the normal or eubiotic conditions, and you will see this epithelial lining, and then there will be a thick mucus layer, and a lot of microorganisms, healthy community, would be on that cellular location. So thick mucus, we call it tight junctions, diverse microflora, and these are all good properties. But as soon as just the opposite of the eubiosis, which is called dysbiosis, which is the abnormal condition. Maybe you are under stress or some infection agents And so what happens in these biotic conditions, sometimes this mucus layer disappears, like a healthy mucus layer you have, that disappears, and then sometimes this mucus, the epithelial tight junction called become leaky. You heard about leaky gut? That condition could be very detrimental, and a lot of microorganisms grow and then can pass through this barrier very easily, and that's called dysbiotic condition. Okay, and in order to understand what happens, the microbial community forming biofilm, and that really try to maintain the eubiotic conditions or eubiosis. Okay, so if you look at the biofilm formation in the gut, again, remember this morning I keep telling you, no matter what surface, abiotic or biotic, the mechanism is very simple or similar. Okay, so they also start same way on the mucus layer. These are epithelial layer. This is the mucus layer. This is the lumen. So you have planktonic cells, right? They find a group, they form a microcolony, and eventually they mature, and then they disperse, right? And so this biofilm formation is pretty similar as what we talked this morning, right? And then it's dispersed, and then can start again. And so similar thing happening in the gut, okay, when they're forming biofilm. And that's how a lot of our microbial community maintained in a positive way in the gut. So don't think that it is a negative, unless you have microorganisms which are causing severe damage to your gut. And that's the dysbiotic condition diseases. So this is a natural or normal process, right? Let's look at on the left, you have a nice thick mucus layer. That's the healthy conditions, right? Presence of commensal microorganisms on the left. And so this is a mucus layer, these are the microbiota. Okay? What happens when your mucus layer becomes thin or thinner and disappears? When do you lose your mucus layer? Anybody? At what conditions people lose their mucus layer? Because mucus layer is very thick, it maintains the health of the gut. Okay? And that's where a lot of this microbial community reside, and they maintain a healthy environment, gut environment. Okay? I think you raised a hand. You want to answer? Okay, so the answer he's saying, when someone get infected by Helicobacter pylori, the infectious agent, that can also affect your mucus layer, right? Any other conditions? Excessive? Oh, hydrochloric acid secretion. Okay, that can also affect it, but it could be that you have mucus layer damage. That's why you got more HCl secreted. So it's a reverse order. Any other? Anybody else this side? He represented left side. Oh, okay. pH. Oh. So changes in the gut pH. Okay. Okay. Anybody else? So for example, if you are starving for a couple of days or 2 days or half a day, there is no food for you. When you go back to the hostel today, there is no dinner for you. Then we decided that, okay, there is also no food tomorrow morning. What will happen to your gut? What do you feel? Come on, you need to wake up. I know it's after lunch. Usually it's pretty sleepy, right? I know all of you feeling it. So what did I ask you? So if there is starvation, right? That means you did not eat. So what's your microbe going to do in your gut? They're still there, right? They need food. What are they going to do? They are all going to eat your mucus, right? Mucus is a good food. That's what this microbial community on the left. Okay, so gradually, so the starvation, that's one of the major factors. Nutrition, right? So all of your microbes, they're going to chew up your mucus. So eventually mucus is going to go away, right? So what happens? These microbes or pathogens, if some of they are opportunistic or invasive, they're going to get in and cause problem. And so this is what you see, absence of commensal biofilm. Commensal biofilm forms when you have a nice mucus, and that biofilm is gone. Now I'm talking about healthy, desirable biofilm. Not negative biofilm, right? These are good biofilm. We need them. And if your mucus layer is gone, they are going to form biofilm. And then you are going to missing out all your microbial community, because the diverse microbial community in the biofilm maintain our gut health. That's why we are healthy. There are a lot of conditions, hydrochloric acid, achlorhydria, also stress. If you don't sleep and during exam period, a lot of times you stay up all night, your HCl secretion mess up. A lot of those factors play important role in your gut conditions. One condition is called achlorhydria. Achlorhydria is when your stomach do not secrete enough hydrochloric acid. That makes you more vulnerable to infection, because a lot of pathogens can pass easily and can cause problems. So these are good examples for biofilm to be maintained in your gut to keep you healthy. So biofilm and disorder, GI disorder. So what we are saying is that under gut disorders, a lot of diseases happens. If the biofilm is gone, good biofilm is gone, then all those bad microbes, they're going to colonize, they're going to invade, and they're going to cause inflammation. Are you with me so far? The good people gone, the bad people get in, and they're going to cause havoc, right? And that's what happens. A lot of those diseases, IBS, irritable bowel syndrome, inflammatory bowel disease, Crohn's disease, and here also achlorhydria, and a lot of those diseases happen. And these are the microorganisms which are present in our gut. Normally, they are not doing any damage, but when the mucus layer gone, your good microbial community disappear, and they can be invasive, and that's when they can cause a lot of this problem. Okay, in this slide, what you are seeing looks pretty busy. This is saying the number of natural microflora, and this is the different parts of the colon, and this is what you are seeing, natural microbial community, how it keeps us protected. Okay, as soon as you disrupt them, they are in big trouble. So here example, this one, let me see, this one just showing this community, see these organisms and how they are maintained, and these are the biofilm matrix and the bacterial cells inside. This is actual colonoscopy picture, okay, how the biofilm works. But in the disease conditions, okay, so this one, this picture, these are actually very, again, new studies. And so here they are looking colonoscopy before and after jet washing, okay. And here before you can see how red it is, ileum, and this is in cecum, and they try to say if it's biofilm produced by the harmful pathogens or organisms, you want to clean it up, and they clean it up after washing. And so some disease conditions, they are trying to find out that whether this biofilm produced by these undesirable or pathogenic organisms, can it be washed or removed physically inside the gut. Okay, and so that's the actual implication of the biofilm in causing diseases. So of course the inflammatory bowel disease, I don't want to get too much detail, but the whole idea being is that the microbial biofilm, loaded by undesirable organisms, not by natural organisms. So in this slide actually showing these different types of bacteria, viruses, protozoa, fungi, they are part of those inflammatory bowel disease conditions in the gut. Okay. Also the colorectal cancer and inflammatory bowel disease, these are some of those that how this work together within the gut system. The biofilm and the colorectal cancer associated, if you have a cancerous condition, right, and then the biofilm produced by undesirable organisms, and that can lead to also cancerous condition. Okay, so this clearly shows that biofilm in the gut play an important role. In a positive side, it can help you maintain healthy gut conditions, and if in the negative side, it can cause serious disease and as well as the cancer. So there are a lot of studies actually in progress on this specific topic. And so I know we are getting close. So I know I will not be able to finish it, but one of the questions we started asking is that biofilms role in pathogenesis. I already answered some of those questions, but I want to ask a very specific question. If you take a biofilm cells, say for example in a laboratory, you have a petri dish, you are growing bacteria, right, and they are forming biofilm. So one question we wanted to ask, are these biofilm forming cells, which are the sessile cells, right? And if you take a planktonic cells, do they have any difference in the pathogenesis? Are you following me? Because we keep asking, the planktonic cells, they eventually form, right, go through biofilm, eventually make sessile cells, some population become dormant, some become persister, some could be VB and C, right? And now they can come back to planktonic. So you wanted to look at, are those sessile cells, are they pathogenic as planktonic cells? That's the question we want to answer, and I think I will follow up tomorrow. It will be a little bit overlap. So our study would be Listeria monocytogenes as a model pathogen to study biofilm mediated pathogenesis. We actually designed that experiment to ask that question, to answer that question. Okay, I will end it here, and next 2-3 minutes, if you have any questions, go ahead. Can you give him the microphone?
Sir, actually, I have the question about that persister cells. Actually, how are they forming? I mean, how are they actually forming?
How they are forming?
Yes.
That's what I was trying to tell you, is that these cells are responding differently to the stress, right? Okay. And so the toxin antitoxin function.
Yeah, about that system, I didn't quite get that. And toxin antitoxin system.
That's why I'm saying you need some read up more. Okay, let's say the toxin, whenever you say toxin, what does toxin means to you?
Harmful substance.
Toxin, any kind of toxin. So as soon as you see toxin, that means its purpose is to damage, right? Yes. Whether it's a membrane or enzyme or protein or any of those stuff. So when the cells undergoing persister phase, the toxin part, one of those enzymes which actually degrade that messenger RNA, and that RNA actually codes for enzyme that may be required for the bacteria to become a healthy cell, living cell. So it can go through division. Okay, but if that enzyme is arrested or inhibited, the cell loses that function, therefore it cannot be metabolically active. You see this one? And so that may be the beginning stage of their going through the persister phase. Okay, means so it's metabolically less active.
So they actually have a negative role in persister cells. Actually have a negative role, you know.
Persister cells from the bacterial side is good because they want to survive. They want to persist in that environment. But from our side, from the health side, it's negative, right? We don't want them to be there. We want to kill them because otherwise, you know, you are treated with antibiotics, and these are not antibiotic resistant, but still maintained. And as soon as you stop giving antibiotics, they flare up. They come back to life, they cause the disease again, which is not desirable, which you don't want. So normally doctors practice, I know besides some of the strategies they gave you, or I mentioned, normally what they do, if they realize that we are creating these persister cells, then they can stop the antibiotic treatment for a while and let them build up again, and then give a higher doses of antibiotics to kill those cells so that we can avoid the persister phase.
So we are actually increasing the effect of antibiotics to kill the persister cells.
Yeah, it starts with antibiotic. So we're trying to show you the sublethal stress also, right? Anytime you are not maintaining that level or concentration, always some population going through this persistent phase. Okay?
Thank you.
Any other questions? No? But bring some questions for tomorrow. This is my task. So we'll have plenty of time right before you start lecture or during the break, right? Ask. I want to hear some questions from you. You cannot stay silent. You already participated this morning. You told us your hobbies, right? Or what you are studying. So we will hold you accountable. Otherwise you have to sing or dance, whatever hobby is you mentioned. That's part of your punishment. You got to come here and dance, or whoever said singing, right? I will make you do that if you don't bring a question. All right, I will see you tomorrow.
Sir, wait, I have a question.
Okay, go ahead.
Sir, there was this beautiful graph showing resistant cells, persistent cells, and susceptible cells that you showed in your slides.
Yeah, yeah.
So I just want to ask that how do we differentiate exactly between persistent cells and resistant cells? Because eventually persistent cells will become resistant.
No, don't call resistant. They are not resistant. If you call it resistant, they are not persistent.
Sir, so how do we differentiate exactly between persistent and resistant? Because we are giving antibiotics, even resistant cells are staying there and persistent cells are also surviving. So it feels like there is a overlapping thing in between.
If you have to design an experiment in the laboratory, right, you know how to count cells, right? And so if it is a resistant cell, if you started with 10 to the power 8 cell population, at the end of antibiotic treatment, you are still going to get 8 to the power 8 cells. So you have to count the cells to get this population. If it is sensitive, they are going to die, right? Nobody is going to be there. If it is like a tolerant, they will go for a while. So you got to follow that graph though, how they over time, you will count the cells. And so the persistent cell will be the low number cells. They are at the bottom, they are still maintaining. If you remove the antibiotic, they come back. Resistant, we know they are going to be maintaining, right? But this persister, they are going to be maintaining. You withdraw the antibiotics and you can see this coming back. Are you getting it? Dead cells are dead. So you can design an experiment, you can find it. So that data, the graph I was showing you, essentially these are signifying bacterial numbers. And from the number or counting, you'll be able to pick up the population which is going through it.
Got
The
it, sir.
staining and everything else, they're going to be same. They're going to be maintained the same.
Thank you, sir.
Okay, thank you, Professor Bunia. Now we have another speaker from our university, Professor Antaresh Kumar, who is head of the Department of Biochemistry. And Dr. Antaresh joined this university in 2020. Before that, he worked as a lecturer in JP University of Information Technology, Solan. Then he moved to Central University of South Bihar, and he joined here in 2020. And he was promoted as professor in 2023. He received DST Young Scientist Award, CSR Research Intern Award. Then there are plenty of other awards which he has received. He has been actively engaged in research in the field of infectious disease. I know him personally also. He is working on Candida. So that is his topic also. He has 54 peer-reviewed papers in his credit in international journals of repute, 27 book chapters, and has authored one book. He earned a total citation of 1754, which at H index of 15 and I-10 index of 21. He has completed 3 extramural research projects and supervised 2 PhD students, and many more are under his supervision at this university. So I welcome Dr. Antaresh for his lecture.
So thank you. Am I audible? So thank you, Professor Gunjan Goyal, for the brief introduction. After this bacteria-related biofilm infections, we'll move to the fungus first. So the topic for today lecture I choose related to the role of biofilm in pathogenesis and drug resistance in Candida albicans. So today's lecture basically contains three major parts. One part is related to the Candida, which is the opportunistic fungus, and what is the prevalence of this fungus in the human and other species. Second part is biofilm, their contributory role in the pathogenesis. And then last one will discuss about the role of the biofilm in drug resistance, as Sir left his topic on the pathogenesis. So I hope I'll address this thing in Candida albicans. So if we talk about the Candida albicans, it is the human opportunistic pathogens which normally reside inside us, and it acts as a harmless commensal inside the body, and when the immunity is decreases inside the decreases, then this commensal fungus becomes pathogen and causes the different diseases like the superficial to the invasive one. So this Candida albicans causes the third ranks of the nosocomial infections. The patients who undergone through the hospital conditions, and there it causes the cross infection with the Candida albicans, and it is considered the third most infected fungus after the Staphylococcus aureus and Pseudomonas. And the mortality rate in such cases, if it is the Candida infect to the patients, has the more than 50% mortality in the hospital cases. This Candida infections causes both acute and chronic infections, and normally if you observe infection in the rainy seasons, that can be infection can be a short time or the long time, can cause the acute as well as the chronic infection. Even in sometimes the fungal infection can cure after taking some drugs, and then after sometimes it regrow on the same site in the human. So it also causes the episodic infections, and the infection can be a superficial or can be invasive in the different parts of the humans. So the major cause of any infection in the bacteria as well as the fungus is to release some virulence factor, which is the major cause of molecule that basically cause the infections. And this virulence factor basically include the mycotoxins, as Sir said, and also forms the biofilm. So the biofilm has a role in the pathogenesis, and the virulence factor induces the biofilm in different microorganisms, including the Candida albicans. But if you take the treatment, the different antifungals against this Candida infection for longer times, then the Candida albicans tends to develop this resistance against the topical antibiotics available in the market, and this is the major cause for the drug resistance nowadays. In the most of the Candida strains becomes resistant against the first line antifungals, which is the azoles. And the beauty of this fungus is it is polymorphic fungus. The Candida albicans is the polymorphic fungus. It exists in actually three forms. One is this oval shape, which is the yeast form we generally called. Another one in between this yeast and the hypha, that is pseudo-hypha form. So this is the pseudo-hypha structure, which is not exact. This is the pseudo-hypha is the between structure of yeast and hypha, in which the hypha is attached with the yeast cell, is called a pseudo-hypha. So this how this Candida albicans grow in the form of yeast in a media or in the normal conditions when it is non-virulent. So normally in a non-virulent condition is the yeast. Once it is gets some environmental changes, then it becomes a pathogenic. So the virulent form is converted into this, sorry. So this Candida albicans becomes pathogenic when this switching from this yeast transition to the hyphal transitions, and then it takes to adhere to the tissue, and then it causes the infections. So this Candida albicans tends to exist in the different morphological structures, and the morphological structure has a relevance in terms of their virulence and non-virulence and causes the infections. There are different species are known that involved in the Candida infection in the humans. So these are the very common species of the Candida that causes the opportunistic infection in the human, and among these is the most common infection is caused by this Candida albicans. The prevalence of this Candida albicans is more than 60 to 70% depending upon the different type of diseases. And nowadays, in addition to the Candida albicans, some strains are Candida glabrata, tropicalis, Paraplopsis cruzii, and the most recent Candida strain is Candida aureus, which is a recent, has the more prevalence of infections in India as well, and the more resistance against the available antifungal drugs. In addition to this common species, there are some less common species also available, and such as this Dublin-cis, Candida Dublin-cis, Candida femata, and other species, and some rare species of Candida albicans also exist that can also cause the rare infection in humans and other. So this Candida albicans can cause the candidiasis at different sites. Suppose it's if it is adhere at the mouth cavity, then it causes the mouth candidiasis and also causes the superficial infections and the cutaneous candidiasis, and a different part of the invasive candidiasis at bone, heart, lungs, and other side of the human body. It also causes the candidiasis at the vaginal candidiasis, which is the most common infection in the female by the Candida albicans, because the male, as compared to the male, female are more prone to this Candida infection due to the hormonal disbalance. And other site of the infections are nails and the penile candidiasis. So this Candida can causes the different site of infection, superficial to the invasive at the different site of the humans. And if you talk about the epidemiology of this Candida infection, so this causes the approximate over the 1 million morbidities cases annually, and 9 to approximate 10 or 1 million mortality overall worldwide by this Candida infections. It is more prevalent to the person who are undergone to the immunocompromised, such as the burn patient and the diabetic patient are more prone to the Candida infection. And third epidemiology, that the cases of the Candida is also increases when the person is either not take the full course of the antifungals or can take the antifungal drug for the long time, thereby the strain, Candida strain develop the resistance, can cause more epidemiology of this. So we have taken some example of this Candida albicans infection, particularly in the burn patients. So if you look at the Candida albicans prevalence in the burn patients, approximate 49% cases are present of the Candida albicans in the burn patient, 33% in the Candida tropicalis, and rest species are very less in the burn patient of this prevalence. Next one, in case of the diabetes, this Candida approximate causes the infection by almost 47%, glabrata 38%, and in the AIDS patient, this Candida is more prevalent because the AIDS patient has very compromised immunity. So the infection rate of the Candida albicans in the AIDS patients are higher relatively to the burn and the diabetic patients. So how the question generate how this fungus or Candida try to enter inside the body, either through the superficial or through the epidermal sites. So for any kind of infection, I told this, the candida has to convert into the non-virulent form to the virulent form. And for this switching, this switching can occur due to the imbalance of the fungal colonization and some alteration in the host environment where the fungus is interact or adhere to that site. So if it is any imbalance between the fungus or the host environment, in that case this non-virulent fungus, which is a commensal present inside food, becomes pathogenic and cause the disease. Another factor is during this disbalance, this fungus can try to change their morphology, can convert from yeast form to the hypha form, that is the virulence form, is another factor that responsible to cause the entry into the host. And the third one is again this, if we are trying to take the antibiotics or antifungal or steroids, if we are taking the steroids for long time, then the mucosal barrier is the more important for this. In that case, this mucosal barrier gets weakened, and in that case, this can get the opportunity, the fungus gets the opportunity to infect the cells. So these are the major factors which are majorly responsible for causing the infection in the human, even though the fungus is present inside our body as a commensal. So what are the mechanisms, how this fungus can penetrate or can adhere to the body, to the epithelial cells? So this is the mechanism behind it. So there are different attachment and the penetration of the candida pathogenesis can be categorized in the three different stages. One is the attachment, third one in the inducing endocytosis, and the fourth one is the penetration. So when this candida cells, either the candida albicans or some other strain of the candida come contact to the epithelial cells, they tends to interact with this human epithelial cell with certain receptors present on the candida cells, such as this EPA1 and the beta glucan. EPA1 is the adhesion proteins that helps to adhere this candida cells to the human epithelial cells. So after the addition, either the candida cells present in the this oval shape or in the hyper form, interaction patterns are little bit different. If it is the candida is this hypal form, in that case this is the receptor called ALS3, and this HWP, ALS3 is the agglutinin-like sequence protein, this is the adhesion proteins. Another one is this HWP is the hypal wall protein, facilitate to bind the candida cells to the human epithelial cells. So this ALP protein, which is the agglutinin-like protein, tends to interact with the E-cadherin proteins of the epithelial cells, and the beta glucans present in the candida cell wall tends to interact with the EPHA2. EPHA2 is nothing but it is the tyrosine kinase receptor which binds to the beta glucans. And the third one is this HWP1 proteins, which is hypal wall protein, that is the adhesion protein, tend to interact with the transglutaminase cross-linking with this epithelial cells. In that way, after the interaction of this fungus, either in the oval shape or the filamenton shape, then it activates this filamentation and causes the endocytosis. And during the endocytosis, this protein, the adhesion protein ALS3 tends to bind to this EGFR or HER2 proteins, as well as also interact to this E-cadherin protein, and another protein present in the hypha, which is SS1, SS1 is this heat shock protein invasion proteins that also interact, this expression is increases, and this interact to the EGFR or HER2. HER2 is this human epithelial growth factor receptor present in the epithelial cell, and in that way it is with this help of this proteins which is expressed in the endocytosis tends to cause the endocytosis in the cells. And after this endocytosis process, by activation of different proteins in the hypha of this candida albicans, then next stage is the penetration. I mean the main infection. So during the penetration of the fungus to the epithelial cells, either superficial or invasive, it tends to activate certain enzymes. These proteins like SEP5, SEP2, these SEP5 or SEP2 protein gets activated. SEP5 basically induce the enzymes that degrade it and also activate the candida lysine, which also damage to the epithelial cell membrane and induce the epithelial immunity. In addition to that, is also releases the lipases or phospholipases. These are the basically proteases that can degrade the cadherin or also degrade the mucins of the epithelial cells. So in that way, the fungus Candida albicans attached and also gets endocytosized at the sites and cause the penetration inside the host under immunocompromised patients. So next is biofilm, because the Candida albicans during this infection, if it is any environmental changes, can cause the biofilm formation, and the biofilm formation is basically a surface associated sessile cell population packed in the extracellular matrix. So there is an extracellular matrix, inside that there is a sessile cell. Sir said the sessile cells are present in the dormant stage, and during this biofilm enhances the quorum sensing. That quorum sensing is also important for the pathogenesis of the Candida albicans, because this releases some mycotoxin or virulence factor responsible for cause the pathogenesis in the microorganism like Candida albicans. It also slower rate of the cells inside the extracellular matrix. The cells which are present have the slower rate as compared to the planktonic cells, has the different metabolic activity. So relatively slower growth, thereby this biofilm shows the having the extracellular matrix. Even the cells are present in the dormant state, can cause the resistance against the antifungals. So if you look at the composition of this extracellular matrix, which are majorly responsible to inhibit the antimicrobial effect or antifungal effect. So ECM is the cell-produced polymeric jumbles of extracellular DNA that cells has a protein, polysaccharide or exopolysaccharides. Depending upon the strain to strain, these components are very, like glycoprotein has 50 to 55%, carbohydrate 20 to 25% in Candida. Lipids are 10 to 12 to 15%, and extracellular DNA is almost 3 to 5% present in this biofilm regions. So these, what are the role of this extracellular matrix present over the biofilm? So the major role of this extracellular matrix in Candida or normally in the microbes, it facilitates the firm adherence. The primary role of the extracellular matrix, because it has the hydrophobic nature, helps to interact the cell to the matrix or tends to bind to the site where it infects. The second key roles of this extracellular matrix is to maintain the structural integrity of the cells, helps in this, and also third one is protect this cells from the harsh environment, because this extracellular matrix are hydrophobic and it protects from the external environment. And next one is extracellular matrix limits the nutrition acquisitions from the surroundings. So it protects the nutrition limiting condition and also acts as a barrier for fungicidal acts. So formation of the extracellular matrix basically causes the resistance, so the antifungal can be not that activates the planktonic cells and cause the resistance in a pathogenic like Candida albicans. So these are the different majorly biofilm producing Candida fungal species, with the Candida aspergillus nidulans, Cryptococcus, Trichosporin, Coccidioides and the Pneumocystis. These are the different fungus can form the biofilm for and causes the different disease in different organisms. So what are the mechanism of the biofilm formation in the Candida albicans? So if you talk about the biofilm formation, initially Thus, the planktonic cell tends to adhere onto the site. So first step is the adherence, where the planktonic cells which are free try to adhere to the matrix or try to bind to the human site or epithelial cells. And then they, after the binding, they try to grow there, they try to multiply at the site. And this takes around, in case of the Candida albicans, around 3 to 11 hours for this adherence and the multiplication. After this, then the next step is the proliferation. Once this Candida cells can nick there, can join, can adhere there in the site, then they try to grow in this to the matrix, and they grow and proliferate in terms of they change the morphology, and also try to synthesize the extracellular matrix. This takes around 12 to 13 hours in case of the Candida albicans, and this stage is called as the intermediate. So if you look at this stain, they have stained this Candida cells with the two dyes. One is this stained with the concanavalin A, and also stained with the one dye. Another dye they have stained with the FUR1A. So this concanavalin can produce the green fluorescence, while another dye is produce red fluorescence. So concanavalin bind to the polysaccharide. If it is the biofilm is form more green, you will see in there as compared to this in case of the adherence to the proliferation stage. And the last one, after proliferation, the more amount of extracellular matrix is formed, and then the amount of this polymorph ECM is formed more. As a result, you will get the more green color, also have some red spots, that is the dormant cells present inside the mature biofilm. And this will take around to formation of the mature biofilm, the Candida albicans, around 22 hours. So in that way the biofilm formations occurs. Next one is the biofilm in Candida albicans. This physical barrier restricts the direct penetration. Biofilm helps in the Candida albicans to escape the neutrophil targeted ROS activation. So biofilm has to protect the host from certain immunological response. So it try to avoid the immunological response if it is the biofilm form in the fungi. Biofilm, Candida biofilm induce some immune evasion proteins like PRA1 and GDP that avoid to the immune response by blocking the activation of the complement system. MSB2 is overexpressed in the biofilm. So this MSB2 that block the host secreted antibiotic peptides. So the function of this particular protein is also expressed while the biofilm is formed, and the function of this particular protein to block the antimicropeptide synthesized during the pathogen interaction during the pathogenesis. And this biofilm is densely colonized, shall produce high concentration of mycotoxin, can induce the pathogen at that site. So these are the different properties of the biofilm. Can also avoid the immune response of the host by expressing certain proteins. What are the factors that affect the biofilm formation? So there are several factors can affect the biofilm formation. As discussed, like if you use the antifungal treatment for longer time, that the biofilm this biofilm formation can be affected. That if you take the more longer time, this can be, the stain can be resistant. Nutrient availability is the another factor. We discussed that nutrient limitation condition is more favorable to form the higher biofilm formation. Which another external environmental condition can also switch the higher biofilm formation. In addition to this, if there is a co-infection with the fungus, can also increase pathogenesis. In that case, the fungus is more virulent because the bacteria tends to release some metabolites that can induce the biofilm formation in Candida albicans. In addition to that, the immune response, I told, there is a certain immune response molecule can also switch, some protein are expressed that can increase the high biofilm formation in Candida. And biofilm has a role in the signaling, particularly for causing the virulence or causing the pathogenesis. So if you talk about the signaling of fungal biofilm, the quorum sensing molecules is influence the biofilm formation in fungus as well as including the Candida albicans. So if it is the quorum sensing molecule is come contact to the fungus, it tends to cause some signaling, and signaling cascade tends to change the virulence, alter the morphological virulence, increase the virulence, cause the apoptosis or filamentation, increase the pathogenesis, and also biofilm formation as also has the biofilm inhibition to protect the cell from the oxidative stress. So this is the one side can be used to inhibit, to protect the, or so basically to develop the quorum sensor inhibitor to inhibit the growth of the Candida infection, particularly the biofilm formation. So different quorum sensor inhibitor can be designed that can regulate all these activities, can regulate the expression of the virulence gene, and that way you can control the fungal infection. Inhibit the biofilm formation if it is the quorum sensor inhibitor is developed against this, and also inhibition of this ALH linked quorum sensing signals. Also increase the antibacterial antifungal activities if it is inhibitor is designed. Also induces the apoptosis causing the cell death of this fungus, and inhibit the biofilm formation, also sensitize these cells for the antifungal treatment. So we have taken the one example. These are the different quorum sensor molecule which is synthesized inside the fungus. One and most important in Candida albicans is this farnesol. So farnesol basically block yeast to filamentous transition at high cell density and promotes the dispersion of yeast cell by inhibiting the germ or hyperformation in Candida albicans. In other quorum sensor like tyrosine has the function that affect the genes associated with the drug resistance. 1,3-glucan is also promote the germ tube formation, antifungal activity, and there is certain quorum sensor molecule inhibit the hyphal formation, filamentation, and also inhibit the autoantibiotic inhibit filamentation in the Candida albicans. I have taken this one example of the farnesol, how the farnesol play a role in the pathogenesis in Candida albicans. So basically the farnesol synthesized inside the Candida albicans by using this malvenate pathway, and this is the intermediate compound like farnesol pyrophosphate. So the farnesol pyrophosphate, majorly the 83%, this farnesol pyrophosphate synthesizes the cholesterol, and later on synthesizes the ergosterol, which is the key molecule of this fungus, and very less amount, this 17% synthesize this farnesol participate in the pathogenesis or the quorum sensing. So this farnesol, if it is present inside the cell, if it is a concentration of farnesol inside the cell is higher, it tends to block the biofilm formation. As a result, the cells are non-virulent, and if it is this farnesol accumulation is higher, it is toxic to the cell, and cell has to release this farnesol through some transporter, and this farnesol tends to then auto regulate or auto signaling to the cell, can synthesize more amount of this, they can synthesize the signal which can increase the pathogenesis and also increase the biofilm. So overall the farnesol play a role in the biofilm formation and cause the pathogenesis, the fungal infection through this quorum sensor molecule. Next one is, rather than the single biofilm formation of the Candida, sometimes if it is more than one microbial species mixed together form the biofilm formation is more virulent, more pathogenesis, and more resistant. So Candida albicans is most commonly isolated bacterial pathogen in the biofilm that can also that can form the biofilm along with the bacterial communities. So this certain sites like oral cavity, ear wound, and UTI, lungs, and other catheter infections, there are multiple microbial populations grow along with the Candida albicans, and this polymicrobial biofilm formation releases some signal molecules microbial interaction and chemical alteration in the environment regulate this polymicrobial biofilms. So the multi microbial molecules can secrete certain signaling molecule that can regulate the polymorphic biofilm, and the polymorphic biofilm is polymicrobial biofilm is more virulent than the single one. So the single Candida albicans is polymicrobial biofilm is more pathogenic than the Candida albicans itself biofilm, and these are the different microbial, basically the bacterial community present, like in the oral cavity infections, Candida albicans along with this Streptococcus mutant, Gordoni, Staphylococcus aureus, Agitobacter biscus, and some other species will found co-infected with, co-formed the polymicrobial biofilm, which are more resistant and more pathogenic than the single one. Catheter and the chronic lung infection, the P. aeruginosa is found with the Candida albicans, and the bulbovaginal, the lactobacillus, basically present in that site, and the Candida albicans along with this can help to each other for causing the more virulent infection. Gut, the Enterococcus and E. coli species is present along with the Candida albicans can helps to the polymicrobial biofilm for more virulent, more pathogenic biofilm rather than the single one. If we talk about the genetics of this fungal biofilm, so that the main genetic, the transcriptional factor which regulate the biofilm formation through hyphal formation and cell surface protein for the adherent. So the transcriptional factors involved that can particularly regulate the hyperformation and also the regulate the expression of adherence proteins. So this certain transcriptional factor gets regulated in the biofilm formation, such as this BCR1, which is the biofilm cell wall regulator protein in Candida albicans, Paraplopsis, and the fumigators. This particular transcriptional factor gets upregulated in the fungal biofilm formation. ACE2, which is the activating transcription of the cell cycle associated gene, gets also upregulated. EFG1 is also upregulated, which play a role into east hyphal switching. So this transcriptional factor is also upregulated in the fungal biofilm formation in Candida and other Candida species. LEA is basically also upregulated in the Aspergillus fumigatus strains. In addition to the transcriptional factors, some other genes can also be upregulated, like particularly in case of the biofilm formation, that multi-drug transporter proteins can also be upregulated with certain extent. If you talk about this Candida albicans has such this CDR1 and CDR2, the ABC transporter genes can upregulated in biofilm formation, and MDR1 is the MFS protein get also upregulated. ERG1 is the ergostrol biosynthesis gene is also upregulated, which is the main components present in the Candida cell wall. In addition to this, some adherence gene I told is also upregulated, like EAP1, ALS1, 2, 3 can also be upregulated in biofilm formation. Amino acid synthesis gene can also be upregulated in biofilm formation, and cell wall biogenesis gene is also gets activated during the fungal biofilm formation in Candida albicans. These are some master transcriptional regulator present in the Candida albicans can regulate the biofilm formation, such as EGF1, TAC1, BCR1, and in addition to those, some accessories regulators are also present. Suppose this, ALS1 and this, these are the genes can be regulate the adherence during the seeding state. EFG1, TAC1, NDT80 regulate the hypha growth formation during this state. RLM1 and JEP1 regulate the extracellular matrix synthesis in the biofilm, and these genes are also regulate the dispersion of the daughter cells during the biofilm dispersion. So this is the overall genes where they are involved in the overall biofilm formation at different stage. For example, this EAP1, ALS1 is also, these are the adherence gene gets upregulated during the adherence state and the proliferation phase, these are different proteins gets upregulated, that or the transcriptional factor like TAC1, EFG1 is upregulated, that can switch the cells from yeast to hyphal formation, and some adherence protein gets also be synthesized or upregulated during the proliferation and the fermentation. Addition state, these are the list of genes can alters, and the biofilm dispersion, these list genes can also be altered, gets upregulated and down. So these are the overall genetics of the biofilm formation. Some genes gets upregulated, transcriptional factor upregulated. Some genes related to the morphology can yeast to hypha can also be upregulated, can convert the cells in yeast to hypha for the biofilm formation. So this is the one research they have discussed this. They have developed this mutant, ALS1 mutant, HYR, which is the hyphal regulatory gene, and also HWP, hyphal wall protein. So they have mutated these genes in the Candida albicans and then tried to observe the biofilm formation with respect to time. So this is wild type. The more biofilm is formed with respect to time. But when they deleted this ALS1, the both genes, then this biofilm formation is affected. HYR is also less biofilm formation as compared to control. And this HYP, this hyphal ball protein, if it is not synthesized as such, almost there is no biofilm formation occurs if it is this hyphal ball protein gets deleted in the Candida albicans. So these genes has a role in the hyphal formation, can affect the biofilm formation in different extent. Another is this is the another protein they have taken in the Candida glabrata strain. So they have observed basically a GPI anchor proteins, and they try to look at the interaction between host to this Candida glabrata cells. So what they does, they have studied this PWP7 or AD1, which are GPI anchor protein present in the Candida glabrata, and these two PWP7 and AD1 is the adherence proteins which interact with the epithelial cells of the host. So what they did, they made the knockout of this PWP7 and AD1, and they checked this is the wild type, and this is the PWP7 or AD1. So as compared to the wild type, this knockout has the less adherence as compared to this. So this AD1 and PW7 has a role in the adherence as compared this AD is relatively has the same effect as the wild type. So with this result, they have concluded this PWP and AD1 has a role in the biofilm, is a role in the adherence. So the cells cannot be adhered to the host tissue and compromise the adherence of the Candida cells to the endothelial cells. They further observed that the expression of this PWP7 or AD which affect the biofilm formation can only is stationary phase are expressed, not these are the stationary phase, the gene are expressed, but these are log phase. This deleted stage has no expression in case of this in stationary phase. So the PWP7 and AD can express only the stationary phase rather than to the log phase. So by deletion mutant had there is no expression, while the wild type has the expression even in the stationary as well as the log phase. So it means this expression is more in the stationary phase, and deleted one has no expression. It means the role of these two proteins are stage specific, can adhere the protein, can basically express in the stationary phase of the growth of Candida. And they have also observed these mutant are defective in the biofilm formation. So this is the PWP deleted mutant and AD. So these are the difference in the biofilm formation. So these two particularly strains, deleted strains adhere, compromise the adherence and also compromise the biofilm formation if it is deleted in the Candida glabrata. Next, what are the immunology involved in the infection of the candida to the host? So this is the host cells. So when it is adhere to the tissue of epithelial tissue of the host, then it invasion causes, and also in that case it releases the cytokines IL-1 alpha, and this cytokine IL-1 alpha binds to the IL-1 receptor, and it also activates this epithelial cells to release the antifungal host defensive peptides, and this host defensive peptides can bind to the toll-like receptor and also activate the different immune response. Ultimately can also activate this neutrophils which can bind to this and engulf this macrophage inside the macrophage to encounter this infection of candida in the host. So in that way the overall the cytokines are activated, the neutrophils neutralize these infections, and also if it is this the filaments enter inside the host can be engulfed into the macrophages, can kill there inside the cells. So this is the overall immunology of the candida infections. There are different class of antibiotics, antifungal drugs are used for the treatment of candida, and most of the antifungal drugs are basically targeted to the membrane, such as azoles. Azoles are considered to the first most commonly used antifungals, which particularly target to the membrane and block the activity of this 14 alpha demethylase, which is the one of the enzyme involved in the ergosterol biosynthesis. Another class of antifungal drug is choline, which also bind to the ergosterol membrane and also thereby change the membrane integrity by affecting the cell permeability. Pyrimidine is the cytosine permease or deaminase uracil phosphotransferase. It is the target and it forms this FDURMP, which inhibits the protein and DNA synthesis. Allylamine is the another class of antifungal which targets to the squalene epoxidate, and it inhibits the squalene epoxidase enzyme, and as a result the accumulation of squalene inside the cell is more, which is toxic to the cell. Echinocandin is the another class of drug which binds to the beta-1,3 glucan synthase present in the cell wall of the candida, and it inhibits the synthesis of this beta-glucan synthase. As a result, it disturbs the integrity of the cell. So if you observe that most of the antifungal is basically targeted to the membrane. So if it is this targeted the membrane, it basically causes the resistance. So if we use this antifungal drugs for the long times, so it causes drug resistance. In that case, there are different strains which generate the drug resistance are available nowadays in the clinical settings, and not only the candida shows drug resistance against the particular drug, but also shows the drug resistance against the multiple type of antifungal drugs, causes the multi-drug resistance. So multi-drug resistance against this different class of drugs that neither share the common structure nor share the common targets. So in that phase, there are different candida strains are available which shows the multi-drug resistance can show no effect of this, no or less effect to the available antifungal drug. So how this fungi basically experience the drug resistance? So if we take the drug either in the sub concentration or we take the not complete the particular course of the drug, in that case the strain tends to feel some stress and trust that fungus or microorganism try to generate some selection pressure against that stress. So the increased prevalence and dissemination of the resistance is the outcome of the natural selection that was be viewed as Darwinian principle. In that way, the fungi tends to experience the drug resistance. What are the different mechanisms which particularly exist in the biofilm forming candida cells? So the mechanism of particularly the candida biofilm cells are little bit different than the planktonic cells. So suppose this candida biofilm forming cells can increase the cell density. If it is the cell density is more, then the more amount of antifungal is required to kill that cells. So as a result, if the cells density is increases inside the biofilm, can show the resistance. Another mechanism is presence of persister cells. So the persister cells are present in the biofilm formation. That cells can have the different metabolic activity, has less effective against this antifungal. Next one is the effect of the biofilm matrix. The biofilm matrix can restrict the entry of the antifungal drug, thereby they limit the antifungal drug to the target, and thereby they shows the antifungal resistance. And the next is alter target by the point mutation. So there are different mutations caused into the different drug sites, particularly in the ergostrol biosynthesis. If it is the mutation is caused, can affect the drug resistance in basically in fungi. So next one is this the increased efflux pump. As we discussed during this genetics, the genes level of the CDR1 or MDR1 is increasing in the biofilm formation, and these CDR1 and MDR1 is basically encodes for the ABC transporters or major facilitator transporters. So more amount of these protein is expressed in the biofilm forming cells can efficiently transport the antifungal drugs if it is penetrate inside it, and can show the resistance. So these are the ergostrol pathway where this antifungal resistance shows the resistance. So in this biofilm formation, these genes ERG1, ERG3, ERG12, ERG11 and ERG2. This is ERG2, 11, 1 and 3. These particular genes gets upregulated, thereby the more amount of this is synthesized. As a result, more ergostrol is synthesized in the cell, will show the resistance in biofilm forming cells. While in normal cases, the azoles is bind here. If it is azoles bind in the planktonic cells here, it tends to deviate the bioergostrol pathway and synthesize more amount of this particular compound, which is toxic to the cells. So in planktonic cells, azoles binds to here and kills these cells, while in case of biofilm forming cells, the upregulation of these genes are upregulated, thereby the more amount of ergostrol is synthesized in the cell wall, can show the resistance against the antifungal. So next one is this efflux pumps. The drugs, the protein of efflux pumps increase antifungal cells that rapidly efflux out, thereby it cannot reaches to the target, showing the resistance is the another mechanism, antifungal resistance mechanism present in the biofilm cells. There are two type of transporters are present. I told is the one in the ABC transporter, which basically you transport the drug inside to outside in the biofilm forming cells by utilizing the ATP. Another one is this MFS pump, which transport the drug inside to outside through the proton gradient. So there are two type of proteins are overexpressed inside the biofilm forming cells, which can rapidly transport or reflex the antifungal drug before reaching to the target, resulting to show the drug resistance. So how do the drug basically transport by ABC transporter proteins? So basically the drug goes inside. This is the drug enter through this phospholipid layer, and it's bind to the transmembrane domain, and this is the drug binding site, and once the ATP hydrolysis causes, then it reflects out. This is the overall mechanism of ABC transporter protein that helps to translocate the protein or reflects the antifungal drug from inside to outside, show the resistance. Next one, the effect of the biofilm matrix. So the biofilm forming cells has the more amount of ECM So this can reduce the penetration of antifungal drug inside the cells. As a result, the cells are more resistant. In that way, cells are basically more resistant to, sorry. So in that way, the antifungal drug cannot penetrate inside the cell. As a result, it shows the drug resistance. So if I conclude the overall study that the biofilm formation in the fungi can generate the quorum sensing, activate the quorum sensing, which causes the pathogenesis. More amount of extracellular DNA, transcriptional factor activates, formation of more amount of ECM, can have the persister cells, and has the polymicrobial species can be more prevalent, more pathogenic biofilm, and also activate the adhesions proteins, and also activate the flex pumps, thereby it shows the resistance and has this post defensive mechanism is also evolved. So if we conclude this lecture, the biofilm formation is basically make some alteration inside the cell, develop more ECM that can produce a help in the pathogenesis and also helps in the adherence, or also help in the drug resistance by several fold, up to 20,000 fold reported in the Candida species. Thank you. So I think this is the first lecture on fungus. So you can easily distinguish the difference, as Professor Gunjan said, with the bacterial as well as the fungal biofilm.
Okay, so thank you, Dr. Antresh.
Thank you.
And I invite Professor Bhunia just to felicitate our speaker.